I would like to call this meeting of the Microbiology Devices panel to order. I'm Dr. Barbara Vanderpool, the chairperson of this panel. I'm a professor of medicine and public health at the University of Alabama at Birmingham, and I have a focus on development and evaluation of diagnostic products that detect infectious diseases. I note for the record that the members present constitute a quorum as required by 21 CFR Part 14. I would also like to add that the panel members participating in today's meeting have received training in FDA device law and regulations. For today's agenda, the panel will provide preliminary input on the potential future reclassification of nucleic acid and serology-based in vitro diagnostic devices indicated for use to aid in the diagnosis of hepatitis B virus, HBV, infection, and/or for the use to aid in the management of HBV-infected patients. Serology-based in vitro diagnostic devices indicated for use to aid in the detection of past, recent, or current infection with human parvovirus, B19, and cell-mediated immune reactivity in vitro diagnostic devices indicated for use to aid in identification of in vitro responses to peptide antigens that are associated with Mycobacterium tuberculosis infection and/or for the use as detection of effector T cells that respond to stimulation by M. tuberculosis agents from Class 3 to Class 2. Before we begin, I would like to remind the public and panelists that this is a non-voting meeting and ask our distinguished committee members and the FDA attending virtually to introduce themselves. Committee members, please turn on your video monitors if you have not already done so and unmute before you speak. I will call your name. Please state your area of expertise, your position, and your affiliation. So I'd like to start with Dr. Kathleen Beavis.
Good morning. Thank you, Dr. Vanderpool. I'm Kathleen Beavis. I'm a professor of pathology at the University of Chicago, and my main foci are tuberculosis and HIV. Thank you.
Thank you. I'd like to call on Dr. Ricardo M. Lahoz.
I am Ricardo Lahoz. I'm the Director of Solid Transplant Infectious Diseases at the University of Texas Southwestern Medical Center in Dallas. I'm also an associate professor of medicine, and I am a transplant infectious disease specialist.
And Dr. Thomas Moore.
Good morning, everybody. I'm Tom Moore, Dr. Tom Moore. I'm an infectious disease physician in Wichita, Kansas. My area of expertise is just general infections.
Dr. Gary Prokop.
Gary Prokop, CEO of American Board of Pathology and professor of pathology at Cleveland Clinic Lerner College of Medicine, specialty in microbiology and infectious disease pathology.
Dr. Kathy Petty.
Good morning, CEO of HealthSpring Global. I'm an expert in infectious diseases and clinical microbiologist.
Dr. Emily Bloomberg.
Good morning. I'm Emily Bloomberg, a professor of medicine at the University of Pennsylvania, where I'm the director of transplant infectious diseases and of the infectious disease fellowship program. And my specialty is transplant ID.
Dr. Camille Cotton.
Good morning. I am the clinical director of transplant and immunocompromised host infectious diseases at Massachusetts General Hospital and associate professor at Harvard Medical School. My area of expertise is within transplant infectious disease. Thank you.
Dr. Angie Caliendo.
Good morning. I'm Angie Caliendo. I'm a professor at Brown. My expertise is in adult infectious diseases and molecular diagnostics for infectious disease.
Dr. Marcus Pereira. Did I say that right?
Good morning. Yes, Marcus Pereira. I'm an associate professor of medicine at Columbia University Medical Center and medical director of the Transplant Infectious Disease Program. And my area of expertise is also transplant infectious diseases.
Dr. Valerie Ng.
Good morning. I'm Valerie Ng. I'm a professor emeritus of the Department of Laboratory Medicine at the University of California, San Francisco. I'm currently the lab director and the chair of the Department of Alameda Health System. I direct the clinical laboratories within this public health safety net organization. Thank you. Oh, I'm sorry, I'm a lab generalist.
And Dr. Charles Chu.
Good morning. I'm Dr. Charles Chu. I'm a professor in laboratory medicine and infectious diseases at University of California, San Francisco. And my area of expertise is virology and molecular diagnostics.
Dr. Nicholas Wensenson.
Good morning. I am a senior investigator and deputy director of the clinical genetics branch at the National Cancer Institute. My area of research is on gynecologic cancers with a strong focus on infectious disease causes of cancer, particularly HBV and other STDs, where we are doing a lot of work on guidelines and a lot of assay development and validation.
Mr. Brad Spring.
Yes, good morning. My name is Brad Spring, and I am the head of global regulatory policy and intelligence at Roche Diagnostics, and I am the industry rep on the panel.
Dr. Roblina Walker.
Good morning. I'm Dr. Roblina Walker, CEO of Omega High Inc., research scientist as well. Area of specialty is microbiology and infectious diseases.
Ms. Jennifer Schwartzat.
Hi, I'm Jennifer Schwartzat, and I am your patient representative.
Dr. Timothy Stenzel.
Good morning and welcome. I'm Tim Stenzel. I direct the Office of IGRA Diagnostics at the FDA. I am a board-certified molecular pathologist, and I have experience in infectious disease, cancer, and genetics. Thanks.
Dr. Uwe Scherf.
Yeah, good morning. I'm Uwe Scherf. I'm the director of the Division of Microbiology Devices here at FDA. And I've been in this position almost 10 years, and I've worked on all of the products we are discussing here today. Thanks.
And Dr. Christian Roth.
Hello, good morning. My name is Chris Roth. I'm the Deputy Director of the Division of Microbiology.
Thank you, everybody. And now I'm going to turn this over to Candace Knowles, the designated federal officer for today's Microbiology Devices panel, and she will make some introductory remarks.
Good morning. I will now read the conflict of interest statement. The Food and Drug Administration, FDA, is convening today's meeting of the Microbiology Devices panel of the Medical Devices Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972. With the exception of the industry representative, all members and consultants of the panel are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this panel's compliance with federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 U.S.C. Section 208 are being provided to participants in today's meeting and to the public. FDA has determined that members and consultants of this panel are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have financial conflicts when it is determined that the agency's need for a particular individual's services outweighs his or her potential financial conflict of interest. Related to the discussion of today's meeting, members and consultants of this panel who are special government employees or regular federal employees have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouses or minor children, and for the purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, CRADAs, teaching, speaking, writing, patents and royalties, and primary employment. For today's agenda, during session 1, the panel will discuss and make recommendations regarding a potential future reclassification from Class 3 to Class 2 with special controls of nucleic acid and serology-based in vitro diagnostic devices indicated for use to aid in diagnosis of hepatitis B virus, HBV infection, and/or for use to aid in the management of HBV-infected patients. The panel during session 2 will discuss and make recommendations regarding a potential future reclassification from Class 3 to Class 2 with special controls of serology-based in vitro diagnostic devices indicated for use to aid in the detection of past, recent, or current infection with human parvovirus B19. The panel during session 3 will discuss and make recommendations regarding a potential future reclassification from Class 3 to Class 2 with special controls of cell-mediated immune reactivity in vitro diagnostic devices indicated for use to aid in identification of in vitro responses to peptide antigens that are associated with Mycobacterium tuberculosis infection and/or for use as detection of effector T cells that respond to stimulation by tuberculosis agents. Based on the agenda for today's meeting and all financial interests reported by the panel members and consultants, no conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208. Mr. Bradford Spring is serving as the industry representative acting on behalf of all related industry. Mr. Spring is employed by Roche Diagnostics Corporation. We would like to remind members and consultants that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the panel of any financial relationships they may have with any firms at issue. A copy of this statement will be available for review and will be included as part of the official transcript. Thank you. For the duration of the Microbiology Devices Panel meeting on September 7, 2023, Dr. Roblina Walker and Ms. Jennifer Swartzat have been appointed to serve as temporary non-voting members. For the record, Dr. Walker serves as consumer representative to the Antimicrobial Drugs Advisory Committee at the Center for Drug Evaluation and Research, CDER. Ms. Swartzat serves as patient representative consultant to the Cellular Tissue and Gene Therapies Advisory Committee at the Center for Biologics Evaluation and Research, CBER. These individuals are special government employees who have undergone the customary conflict of interest review and have reviewed the materials to be considered at this meeting. The appointments were authorized by Russell Fortney, Director for the Advisory Committee Oversight and Management Staff on 7/25/2023. Before I turn the meeting back over to Dr. Vanderpoel, I'd like to make a few general announcements. In order to help the transcriber identify who is speaking, please be sure to identify yourself each and every time that you speak. The press contact for today's meeting is James McKinney. Thank you very much. Dr. Vanderpoel?
Thank you, Ms. Knowles. At this time, I'd like to invite Dr. Timothy Stencil to give some opening remarks. Dr. Stencil?
Thank you, Dr. Vanderpoel. Welcome to all who are joining us today. Your participation and/or your attention to these important topics today and tomorrow are much appreciated. I want to especially thank our panel chair, Dr. Vanderpoel, and all of the distinguished panel members and all of the FDA staff who have worked so hard to make today and tomorrow possible. Today, we examine whether the FDA can safely downclassify the Class 3 tests for hepatitis B, parvovirus, and tuberculosis. Thank you in advance for the panel's input, as well as the input we received from the public. It is the reason for this meeting, and we look forward to hearing from you. Tomorrow, we'll focus on pandemic preparedness, also a very important topic. And again, thanks to all, and I am looking forward to two great days of panel deliberations. Thank you.
Thank you, Dr. Stencil. At this time, I'd like to invite the FDA to start their first presentation. I'd like to remind public observers at this meeting that while the meeting is open for public observation, public attendees may not participate except at the specific request of the panel chair. FDA, you may now begin your presentation.
Hello, my name is Scott McFarland, and I'm a regulatory counsel within CDRH's Office of Product Evaluation and Quality. Today, I will be providing a high-level overview of the medical device classification process, which forms the basis for our discussion today. The purpose of this panel meeting will be to seek recommendations from the panel members regarding the reclassification of 3 in vitro diagnostic IVD devices that are currently Class 3. Specifically, whether sufficient information exists such that the development of special controls, which along with general controls, could mitigate the risk from these devices such that the devices would provide a reasonable assurance of safety and effectiveness and therefore can be eligible for a Class 2 designation. We begin by explaining the different classes of medical devices. Devices are classified based on the controls necessary to mitigate the risks associated with the device type. Class 1 devices are only subject to general controls. Class 2 devices are subject to both general and special controls, and Class 3 devices are subject to general controls and pre-market approval. These regulatory controls will be discussed in greater detail in the following slides. Importantly, a device should be placed in the lowest class whose level of control provides reasonable assurance of safety and effectiveness. Now let's go into more detail about each of the classes. As mentioned previously, Class 1 devices are those devices for which general controls are sufficient to provide reasonable assurance of the safety and effectiveness of the device. Most Class 1 devices do not require FDA pre-market review prior to being marketed. The slide includes a few examples of Class 1 devices. These include elastic bandages, handheld manual surgical instruments, and different culture mediums. There is also an alternative pathway to determine that a device is Class 1. Class 1 devices could also be devices that cannot be classified into Class 3 because they're not life-sustaining, life-supporting, or of substantial importance in preventing impairment of human health, and they do not present a potential unreasonable risk of illness or injury. And these devices cannot be classified into Class 2 because insufficient information exists to establish special controls to provide reasonable assurance of safety and effectiveness. As previously mentioned, general controls are basic requirements that apply to all medical devices and are outlined in the Federal Food, Drug, and Cosmetic Act. Some examples include ensuring the devices are not misbranded or adulterated, following good manufacturing practices, meeting establishment registration and device listing requirements, and adhering to reporting and record-keeping requirements. Class 2 devices are those devices which cannot be classified into Class 1 because general controls by themselves are insufficient to provide reasonable assurance of the safety and effectiveness of the device, and for which there is sufficient information to establish special controls to provide such assurance. Typically, Class 1 devices require a pre-market notification, generally referred to as a 510(k) submission, prior to being marketed in the U.S. Examples of Class 2 devices include intravascular administration sets, for example, syringes, nucleic acid-based IVDs for the detection of Mycobacterium tuberculosis complex, and endoscopes. There are many types of special controls, but some examples include analytical and clinical testing, sterilization validation, and device-specific labeling requirements. These special controls, in combination with the general controls previously described, provide reasonable assurance of safety and effectiveness for Class 2 devices. Companies must include information within their 510(k) submissions demonstrating how the special controls for the specific device type are met. Class 3 devices are those which cannot be classified into Class 2 because insufficient information exists to determine that general and special controls are sufficient to provide reasonable assurance of the safety and effectiveness of the device, and the devices are life-sustaining or life-supporting, or are of substantial importance in preventing impairment of human health, or present a potential unreasonable risk of illness or injury. Class 3 devices typically require pre-market approval through a pre-market approval application, or PMA, prior to being marketed. Examples of Class 3 devices include breast implants and IVDs for the detection and differentiation of human papillomaviruses. Here is a flowchart that walks through the general decision-making process for each of the classes that was just discussed. We start with determining whether general controls are sufficient to provide reasonable assurance of safety and effectiveness. If so, the device can be appropriately regulated in Class 1. If not, we ask whether there is sufficient information that allows us to be able to develop special controls that, in combination with the general controls, provide reasonable assurance of safety and effectiveness. If so, the device can be appropriately regulated in Class 2. If not, then it would be Class 3 if the device is life-supporting or life-sustaining, or if it is of substantial importance in preventing impairment of human health, or if it presents a potential unreasonable risk of illness or injury. If the device is not life-supporting or life-sustaining, or of substantial importance in preventing impairment of human health, and does not present a potential unreasonable risk of illness or injury, then we end up back at a Class 1 designation. What we asked in the panel today is to provide input and recommendations as to whether 1. Hepatitis B virus antigen, antibody, and molecular assays, 2. Parvovirus antibody assays, and 3. Microbacterium tuberculosis interferon gamma release assays should be reclassified from Class 3 into Class 2. The input should include an identification of the risk to health presented by each device type and a discussion of whether each device is life-supporting, life-sustaining, of substantial importance of preventing impairment of human health, or if the device presents a potential unreasonable risk of illness or injury. The panel will also be asked to discuss whether sufficient information exists to develop special controls and what those special controls should be that, in combination with the general controls, would provide reasonable assurance of safety and effectiveness for each device type. Following this panel meeting, the FDA will consider the available evidence, which includes the input received from this panel and the public, and consider potential future reclassification of the device types discussed today. I hope this has provided you with a sufficient background to set the stage for the forthcoming discussion. Thank you for your time and attention.
Hello, today I will present on the potential hepatitis B virus HBV device reclassification. HBV represents a substantial public health burden. CDC estimates that chronic HBV infection in US affects between 580,000 to 1.17 million people. Because HBV infection can be asymptomatic, many individuals are unaware of their HBV infectious status. Approximately 95% of adults with acute HBV infection recover completely, whereas 5% of adults develop chronic HBV. Infants born to women who are hepatitis B surface antigen positive are at high risk of HBV infection. In the absence of treatment, infants infected with HBV have a 90% risk of progression to chronic HBV, and up to 25% of infants who acquire chronic HBV infection will die prematurely. Chronic HBV infection increases the risk of developing liver damage, liver cancer, and liver failure. In addition, HBV can be reactivated in patients receiving immunosuppressive therapies, resulting in serious risk of liver failure or liver-associated death. For these reasons, diagnosis of HBV infection with HBV antibody and antigen tests is essential to ensure that patients are linked to appropriate care. Current CDC guidelines recommend screening for all adults aged 18 years and older for HBV infection at least once during their lifetime. This includes testing for surface antigen antibodies, and core antigen antibodies. Pregnant people should also be screened with a surface antigen test during each pregnancy. The table shows the different interpretations: acute infection, chronic infection, resolved infection, immune due to vaccination, and others, and the follow-up actions to take based on the results of HBV antigen and antibody tests. HBV antigen assays have the following intended use, where the assay may be used to screen for hepatitis B infection in pregnant women to identify neonates who are at risk for acquiring hepatitis B during the perinatal period. Assay results, in conjunction with other laboratory results and clinical information, may be used to provide presumptive evidence of infection with HBV, the state of infection or associated disease not determined, in persons with signs and symptoms of hepatitis, and in persons at risk for hepatitis B infection. Qualitative antibody assays have the following intended use, where the assay may be used as an aid in the diagnosis of adults with acute or chronic hepatitis B infection, and in the determination of the clinical status of HBV infected individuals in conjunction with other HBV serological markers for the laboratory diagnosis of HBV disease associated with HBV infection. This assay can also be used as an aid in the differential diagnosis in individuals displaying signs and symptoms of hepatitis. Quantitative surface antigen antibody assays have the following intended use, where the assay may be used as an aid in the determination of susceptibility to HBV infection for individuals prior to or following HBV vaccination, or where vaccination status is unknown. Assay results may be used with other HBV serological markers for the laboratory diagnosis of HBV disease associated with HBV infection. A reactive assay result will allow a differential diagnosis in individuals displaying signs and symptoms of hepatitis in whom etiology is unknown. The detection of anti-HBs is indicative of laboratory diagnosis of seroconversion from HBV infection or from vaccination. The risks to health for HBV antibody and antigen tests include false negative results. These may result in misdiagnosis of infected patient, incorrect HBV infected status determination, including delay, failure to perform additional diagnostic procedures, and linkage to appropriate care. Unnecessary testing in pursuit of another potential cause of hepatitis, and potential transmission of HBV to others. A way to minimize potential sources of false negatives is for the assay to have optimal sensitivity. The risks to health for HBV antibody and antigen tests also include false positive results. These may result in misdiagnosis of infected patient, incorrect HBV infected status determination, including unnecessary diagnostic procedures, missed opportunity for vaccination, and psychological stress to the patient. A way to minimize potential sources of false positives is for the assay to have optimal specificity. When the patient is diagnosed as HBV infected with the antibody and antigen assays, they are then tested with HBV DNA to guide treatment decisions. Because current HBV treatments are lifelong, patients receive regular HBV DNA testing. The goal of HBV treatment is sustained suppression of HBV replication, also known as undetectable HBV DNA. This leads to improved liver enzymes, loss of hepatitis B E antigen with or without detection of hepatitis B E antigen antibodies, and improvement in liver histology. HBV molecular assays have the following intended use, where the assay may be used as an aid in the management of patients with chronic HBV infection undergoing antiviral therapy. The assay can be used to measure HBV DNA levels at baseline and during treatment to aid in assessing response to treatment. The results from the assay must be interpreted within the context of all relevant clinical and laboratory findings. The risks to health for HBV molecular tests include false negative, falsely decreased results, incorrect interpretation of test results, failing to correctly operate the test. These may result in withholding or premature discontinuation of HBV antivirals, potential transmission of HBV to others, potential for other unnecessary medical procedures to investigate other causes of liver disease. A way to minimize potential sources of false negatives is for the assay to have optimal sensitivity. The risks to health for HBV molecular tests also include false positive, falsely elevated results, incorrect interpretation of test results, failing to correctly operate the test, and these may result in administration or continuation of unnecessary antiviral treatment, psychological stress to the patient. A way to minimize potential sources of false negatives is for the assay to have optimal specificity. Special controls could potentially be developed to mitigate the risks of false positive, false negative, incorrect results interpretation, and failure to correctly operate the device. The goal is to maintain consistently high performance across devices with similar intended uses and for individual devices of the total product lifecycle. The following slide shows the questions for the panel to discuss. I will read each one. 1. Please comment on whether you believe FDA has identified a complete and accurate list of risks to health presented by the following devices: qualitative HBV antigen tests, qualitative HBV antibody tests, quantitative anti-HBs tests, and/or quantitative HBV molecular tests. Please comment on whether you disagree with inclusion of any of these risks, or whether you believe any other risks should be included in the overall risk assessment of the devices listed above. 2. Please discuss potential mitigation measures, controls that FDA should consider that could mitigate each of the identified risks. 3. Based upon the information presented and future discussion at this panel meeting, please discuss whether, based on the available information, the panel believes FDA should initiate the reclassification process for these devices from Class 3 to Class 2, subject to special controls. 4. Currently, there are no FDA-authorized tests for the detection and quantitation of hepatitis B surface antigen. Please discuss appropriate intended use for such a device, potential risks associated with that intended use, and whether mitigation measures, special controls could be developed that, in addition to general controls, mitigate risks to health. Thank you.
We will now proceed to the open public hearing portion of this meeting. Public attendees are given an opportunity to address the panel to present data, information, or views relevant to the meeting agenda. Ms. Knowles will read the open public hearing disclosure process statement.
Both the Food and Drug Administration, FDA, and the public believe in a transparent process for information gathering and decision-making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationship that you may have with any company or group that may be affected by the topic of this meeting. For example, this financial information may include a company's or a group's payment of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking.
Thank you, Ms. Knowles. FDA has received 4 requests for the open public hearing portion. Each speaker will be given 5 minutes to speak, and we will begin with Dr. Yasmin Ibrahim.
Hello, this is Yasmin Ibrahim, a public health program director at the Hepatitis B Foundation, located in Doylestown, Pennsylvania. Dear committee members, currently only laboratory-based hepatitis B diagnostics are approved, meaning a blood draw must be taken and analyzed in a lab. This means multiple visits are needed to diagnose hepatitis B, which is a significant barrier for those groups that are most impacted by hepatitis B, like communities of color, foreign-born communities, the LGBTQ community, and people who use drugs. Blood draws are associated with increased cost, can be logistically difficult to perform in low-threshold care settings such as syringe services programs or community-based care, and impacted communities are often hesitant to draw blood. Additionally, there is lag time, which increases loss to follow-up while waiting for days for test results, particularly in community screening settings where a large percentage of screening of high-risk people takes place. In 2023, the Centers for Disease Control and Prevention recommended that all adults aged 18 years and older should be screened at least once in their lifetime using a triple panel test. This recommendation aligns with the U.S. Department of Health and Human Services plan to eliminate hepatitis B in the United States. This new recommendation provides an ideal opportunity to improve hepatitis B diagnosis rates in the United States, and we strongly believe that hepatitis B point-of-care rapid diagnostic tests are necessary to implement these recommendations and could be a catalyst to scaling up hepatitis B screening. Point-of-care rapid diagnostic tests will allow us to address current challenges and improve hepatitis B diagnosis across the United States to move infected people into life-saving care and treatment and play a critical role in reaching the goals of viral hepatitis elimination in the United States. Here are a few highlights about the burden of chronic hepatitis B in the United States. Up to 2.4 million people in the United States are chronically infected with hepatitis B, and without timely diagnosis and treatment, 25% will die prematurely from liver cancer. Currently, up to 70% of people with chronic hepatitis B infection in the United States remain undiagnosed. At the same time, we are seeing a troubling increase in new acute hepatitis B cases in many states, largely associated with the opioid crises among people who use drugs, which is another population that has limited healthcare access. Hepatitis B disproportionately impacts Asian Americans, Pacific Islanders, Native Hawaiians, and people of African origin. These primarily immigrant and refugee communities face unique barriers to prevention and care and hold their own unique set of social determinants that lead to reduced access to healthcare, stigma and discrimination, and serve as barriers to diagnosis. Access to rapid diagnostic tests for hepatitis B would reduce delays and complexity due to phlebotomy and laboratory testing. Additionally, there are benefits to early diagnosis of chronic hepatitis B. Hepatitis B damages the liver over time. Even without alarming symptoms, hepatitis B can lead to cirrhosis and liver cancer. Additionally, recent research indicates that hepatitis B viral DNA integrates into the host genome early and consistently throughout the infection. Access to timely diagnosis is an essential step in managing hepatitis B and improving health outcomes of those impacted by the disease. Upon diagnosis, individuals with chronic hepatitis B must be evaluated and advised on a proper management plan. Antiviral therapy controls viral replication to help prevent hepatitis B-related liver disease like cirrhosis, liver cancer, and premature death. Similar to people living with HIV/AIDS, people living with chronic hepatitis B are expected to live long, healthy lives if they gain access to timely care. To this end, we respectfully request that the Food and Drug Administration consider the reclassification of hepatitis B testing from the current Class 3 to Class 2. Thank you.
Thank you. We'll now hear from Dr. Sue Wong. Dr. Wong, go ahead.
Great, thank you. Let me just pull up my slide. Sorry, of course I can't find it now. That's fine, I'll go ahead. Dear committee members, thank you so much for having me. My conflict of interest include a Gilead grant that funds my institution for a research project that I have. I'm a practicing internist and I'm director for the viral hepatitis programs and the Center for Asian Health at the Cooperman-Barnabas Medical Center in New Jersey. I'm also living with hepatitis B, and I'm former president of the World Hepatitis Alliance, which is an NGO dedicated to harnessing the power of people living with viral hepatitis to achieve its elimination. I also serve as senior advisor of global health to the Hepatitis B Foundation. In 2016, WHO set forth the goal of eliminating hepatitis B as a public health problem by 2030. This is an achievable milestone because we have tools to eliminate hepatitis B. We have ways of testing people, we have effective vaccines, and we have treatments to suppress the virus to prevent end-stage liver disease, liver cancer, and prevent transmission. And hepatitis B elimination would be a very impactful achievement. Globally, hepatitis B is the most common chronic bloodborne infection. There are 350 million people in the world living with hepatitis B. That's 7 times more than those living with hep C and almost 10 times more than people with HIV. Yet, only 10% of these 350 million have been diagnosed, the vast majority of people largely unaware of having this infection. The need to scale up testing is one of our largest gaps to achieving elimination. Without being diagnosed, people living with hep B cannot get care or treatment to reduce the risk of liver cancer or the risk of transmission. In the US, up to 2.2 million people are living with hep B, and about 2/3 of them remain undiagnosed. More than 50% of people living with hep B in the US are Asian American Pacific Islanders, and 12% are African in origin. It is well documented that many of these communities face significant barriers to accessing healthcare in our country. Down-classifying hepatitis B assays would be instrumental to increasing access to testing, and it would be an enabler for the US to achieve our goals set forth in the HHS Viral Hepatitis National Strategic Plan. It would allow for more manufacturers to seek approval for tests, and it would expand our testing options, which is much needed. Point-of-care rapid diagnostic tests for hepatitis B surface antigen have existed for years and are approved for use in other countries. At least 2 that exist are already on the WHO pre-qualified list, and others also have high sensitivity and high specificity. These tests are used around the world and have increased the reach of testing efforts. Many cost as little as $1 a test and do not require phlebotomist or expensive lab equipment. Yet none of these are FDA approved and thus not available in the US. There are numerous publications that show the effectiveness of point-of-care tests and also their use in various settings. Point-of-care tests allow for healthcare in low-barrier settings, such as community clinics, health fairs, and other community-based settings. These are especially important in populations that face significant hurdles to accessing medical services, maybe due to geography, lack of insurance, lack of transportation, childcare, or work that doesn't allow them to leave during office hours, or limited English proficiency. Rapid diagnostic testing also importantly allows for testing and diagnosis to occur at the same time. Patients know the results of the test shortly after being tested. There's no prolonged anxiety from waiting, and they do not have to arrange the logistics of getting the result, which could be another appointment. They have to take a time away from work, a phone call that they miss, or a letter that they may not be able to understand. This is critical for patient-centered care. Once people know that they have hep B, they can begin taking care of themselves. They can educate themselves, they can make an appointment for hepatitis B care, get additional testing to see if they need treatment, which will include the hep B DNA test and other liver studies. And they can begin protecting their liver. They can limit alcohol use and herbal medications, and they can begin discussing with their loved ones, including family members and sexual partners. One of the many struggles currently with testing is reaching people to notify them of their test results, and point-of-care tests would really help with the loss of follow-up. Additional HBV assays, and maybe we could change the terminology to tests as was done for hep C, include qualitative and quantitative hep B antibody tests and quantitative hep B molecular tests. Down-classifying these as well will help with identifying whether people are immune to hepatitis B. And if they're not immune, they should be vaccinated following the recent CDC updated recommendations for universal vaccination. Down-classifying the hep B DNA test will be helpful as this is necessary for evaluating the need for antiviral treatment. We also anticipate that there will be point-of-care hep B DNA tests developed in the near future. FDA has downregulated. Okay, I just have one sentence. FDA has downregulated hep C and HIV tests to class 2, and we believe that the safety and effectiveness criteria that allow for these also apply to hepatitis B tests. In conclusion, many of us in the hep B provider, patient, and affected communities have been long awaiting this down-classification of hep B tests. We believe this will play a critical role in expanding hep B testing and close the gaps in hep B diagnosis and care so that we can make hep B elimination a reality. Thanks.
Thank you. And now we'll hear from Dr. Diana Zuckerman.
Thank you so much. Can you hear me?
Yep.
Great. I'm Dr. Diana Zuckerman. I'm president of the National Center for Health Research. Our nonprofit health think tank, public health think tank, was founded in 1999, and we focus on the safety and effectiveness of medical products. We do not accept funding from companies that make the products that we evaluate, so I have no conflicts of interest. My expertise is in epidemiology and public health, and I previously was a researcher at Harvard and Yale and a congressional investigator in the U.S. House of Representatives and the Senate, where I evaluated the impact of FDA policies. I've published numerous articles regarding the impact of FDA policies, and I'm a founding board member of the Alliance for a Stronger FDA, which is a coalition of industry and nonprofit organizations that work together to ensure sufficient resources for FDA to support its very important work. So I want to start by thanking you, all of you, for your important work today. And I'm going to speak briefly this morning about your goals of the meeting today. As you know, these assays have been designated as high-risk Class 3 devices that patients depend on for accuracy, sensitivity, and specificity, and you've heard about that this morning. I've conducted numerous studies on device recalls and have found that Class 3 devices are much less likely to be recalled than Class 2 devices because the standards for getting them on the market is much lower for Class 2. The FDA does not refer to Class 2 devices as approved. They are referred to as cleared for market. And there's a good reason for that distinction. And that's what concerns me today. I am not an expert on the assays being discussed today, but I have spoken with experts from medical schools and the NIH and learned that these assays already exist, as you've heard already. And so what's the need for changing the category to Class 2? Lowering the standards in an effort to get them to market more quickly without the data needed to know how accurate they are is potentially very harmful to patients, either because of false positives or false negatives resulting in inappropriate treatment. And you've heard from everyone about how important that treatment is. So in conclusion, these assays are very important, and they're going to have an enormous impact on treatment decisions for patients. And although special controls can help improve the accuracy, they aren't the same thing as the standards for a Class 3 device. And for that reason, I respectfully suggest that you think very carefully about whether lowering the classification is really in the best interest of patients. Thank you very much for the opportunity to speak today.
Thank you. And finally, we'll hear from Dr. Robert Gish.
Good morning. Is audio and video okay?
Yep, you're good.
Yep. Thank you so much for having me here. I really want to thank the committee for having this open comment session. I am Robert Gish. I'm a hepatologist. I'm based in San Diego. I'm the medical director of the Hepatitis B Foundation. I'm also the medical director of a local organization called the Asia Pacific Health Foundation. My disclosures are consultancies for Gilead and a number of other hepatitis B companies in the therapeutic space. I'm very committed to hepatitis B cure and also work in the diagnostic space. I want to give just a little bit of history to this down classification. I was honored to be invited and visited the FDA over a decade ago with Dr. Robert Perillo and Dr. Tim Block asking about the steps for down classification. We invited 2 diagnostic companies to be at that meeting, and one declined to come to the meeting within 24-hour notice, and the other came, but after that meeting, we didn't hear from them again because they were looking at and discussing the barriers of entry of a Class 3 designation for hepatitis B testing. I think this is a very important issue about getting diagnostic companies into this space. I want to give another personal perspective, and that is I've worked on hepatitis B elimination in over 70 countries worldwide. In the last 6 months, I've been in many countries in Southeast Asia, and 2 weeks ago, I spent a week in China working on and surveying what would be the impact of a rapid test in the community. One of the things that's very important in the hepatitis B world is the issue of stigma and discrimination. You've heard from Dr. Ibrahim and Dr. Wong all the important issues that I think are key to this down classification. But part of the reason there's not broad-based recommendations about testing is the issue of stigma and discrimination. And I believe the down classification and bringing rapid tests and point of care tests available to patients and individuals in private settings will be very important. The next step beyond that of testing in a setting where you have a medical provider is at-home testing. And at-home testing can be definitely catalyzed by having a rapid test available. We have apps, we have artificial intelligence, we have many other techniques now to help interpret these tests so individuals can test themselves and make steps individually without necessarily having their hepatitis B tests in a public setting. We believe that stigma and discrimination are also reasons to lead to hepatitis B cure and global elimination. Yes, hepatitis B cure will help decrease liver cancer, cirrhosis, transplant, and deaths. But we want our patients, our individuals in the public setting to be able to work in a number of different occupations. We want people to have normal lives and relationships, and down classification is important. In San Diego, we do screening in a number of immigrant communities, and having to do a finger stick for hepatitis C and then a blood draw for hepatitis B, and then trying to find the patients 1 to 2 weeks later to communicate the results, interpret the results, and bring those results to a linkage to care is a multi-step process and leads to a significant dropout of patients being linked to care. We really want this down classification to impact our patients and our providers in the US, including the pharmacists, who I think will be a key part of rapid test distribution, education, and interpretation. But also this affects the world. In each country where I've met with regulatory and commercial providers, they always ask the question, is your test FDA cleared, or will your test be FDA cleared in the near future? What is the path forward? If we had that green light from the FDA clearance, it would affect not just our patients and providers in the US, but affect individuals globally. I want to thank you for having me here today and allowing me to put this professional and personal note on top of what Dr. Ibrahim and Dr. Wong provided. I think these are very important issues for the down classification. Thank you so much.
Thank you. Does anyone on the panel have any questions for our open public hearing speakers? If you do, please use the raise hand function. Okay, let's start with Dr. Wenceson.
I actually have a question for clarification to the FDA, because we've heard what I think is a little bit of a conflation between point of care testing and the device classification. And I just wanted to hear whether it is correct that these are two separate issues. Like, I think there are class 3 devices that are point of care tests, and there are class 2 devices that are not point. So I just wanted to hear a clarification on that.
Yes, Tim Stenzel. Happy to clarify. Yes, there's nothing that prevents a class 3 device from becoming a point of care or even an over-the-counter test. In fact, the over-the-counter HIV test is a class 3 test. So, I did hear that potential, confusion here, and I'm happy to clarify that. Thank you so much for the question.
Thank you. Dr. Beavis, you're on mute. You're still muted.
Thank you. Yeah, my question was along the same lines, and it's been answered that there's nothing that keeps a point of care from applying as a class 3. So thank you.
Any other questions?
Yes, Marcus Pereira here. Somebody had mentioned that HIV testing is class 2, and maybe we'll be talking more about this later, but can somebody clarify sort of what parts of the HIV testing sort of menu are class 2 and which ones are class 3?
Yes, HIV devices are regulated by CBER. So if there's a I'm not sure if we have anybody from CBER who wants to respond to that today. If not, I can do my best attempt. So my understanding is that over-the-counter tests for HIV are still class 3, but all other tests for HIV are class 2. That is the present situation as far as I understand it. Thank you.
Any other questions? Then I now pronounce the open public hearing session to be officially closed. We will take a break at this time. We're slightly ahead of schedule, so why don't we take a 15-minute break, and that will put us back here at 10 after the hour. So if you will rejoin us at that time, thank you. Welcome back, everyone. It is now 10 after the hour, and I'd like to resume this panel meeting. At this time, before we begin deliberations, does anyone on the panel have any clarifying questions for the FDA? Please use the raise your hand button, and I'll call on you in the order I see your hand come up. Seeing no questions, we're going to move forward, and from here forward, we will focus our discussion on the FDA questions that were provided to us. Panel members, you have copies of these questions in your packet. We will briefly display the questions and discuss them one at a time. Please don't forget that when you're called upon, reiterate your name and your organization to make sure that the transcription can capture who's speaking. So if you'd show the first question, please. Okay, so the first question is to comment on whether you believe the FDA has identified a complete and accurate list of the risks to health presented by the following devices. Please comment on whether you disagree with the inclusion of any of these risks, or whether you believe any additional risks should be included in the overall assessment. So with that, I'll take input from the panel. As I said, please just raise your hand. Does everyone have any thoughts about the risks as described being concerns about false negatives and what happens in those cases, concerns about false positives?
Sorry, Bobby, this is Valerie Ying. I would like to add one more risk.
Go ahead, please.
And the risk is related and it's overlaid, which is testing performed as point of care.
And what do you see that risk as, aside from the performance of the actual assay?
It is sort of a common observation that test performance is not the same when testing is performed as point of care. And that relates to environmental as well as personnel. And so the engineering of these devices must be incredibly strong to mitigate those risks.
And I'm not clear, and I would love for someone from the FDA to weigh in. I know this is the panel discussion portion, but for a clarification perspective, we're not really discussing point of care tests. We're really discussing the risks of moving from class 3 to class 2. And I know that as we described already, the public speakers were focused on point of care tests, but a class 3 can be a point of care test. So that's not really quite to the point of our discussion. But if the FDA would like to hear more about that topic, Dr. Stenzel, maybe you can respond to that.
Yeah, Tim Stenzel. Yeah, so the focus is for this question on are the lists of risks complete and accurate. So while point of care doesn't necessarily, well, just to say that I think the FDA welcomes the input from Dr. Eng that that there could be risks based on certain technologies that need to be included when you look to mitigate risks and as explained that it could be point of care, could also be over the counter relative to central lab. So I think that input is valuable, and we thank Dr. Eng for it.
So I see Dr. Wenceson has his hand raised as well.
Yes, thank you. I think it would be very helpful, and as you move forward with this decision process, to try to quantify these risks as much as possible, and kind of maybe generate like a simple model that allows to input different assumptions, like a certain reduction in sensitivity would have the following impact on a specific indication. Some of these tests are applied widely in the population, some in very specific settings. Some have like direct consequences related to treatment. Others are monitor an active infection. So I think the qualitatively that list is good. It could be expanded, but I think the quantitative pieces of that are very important for these considerations. And it reminds me of some discussions we have on HBV testing and self-sampling, where we see like a small reduction in sensitivity is there's still a net benefit of expanding the reach of testing, but if that's bigger, then there is kind of equipoise, and et cetera. So I think quantification of these risks to me is very important.
And using chair's prerogative, I'll just respond to that to say that I think one of the things that's not on this list of risks is the list of benefits. And I think that risk-benefit ratio is quite an important consideration, which is partly what I think you're getting at, Dr. Wenstenson. Let me call on Ms. Schwartzat, you're next.
Jennifer Schwartzat, patient representative. I wondered what the percentages of false negatives and false positives are in the class 3 testings assays, and also if anybody knows what those same percentages are in the class 2 similar tests that are already on the market.
That's a great question. I'm not sure if anybody has the data.
Well, yeah, this is Tim Stenzel. I might call on one of my colleagues from the office and to see if they can add to this. So no test is perfect, whether it's class 3 or class 2 or class 1, point of care, central lab, or over the counter. It's all about assigning the proper risk and mitigating any risk. Those mitigations can be technical. They can be through training. They can be through labeling. So in all cases, we hope to get the best possible test performance that is possible for a given analyte. And some analytes are more difficult than others. I don't personally have a compilation of the performance. I don't know if anybody from the Office of In Vitro Diagnostics is comfortable enough responding specifically to this question, but Inez has just come on camera. But we do look at the risks versus the benefits, and as has been mentioned, and we do take it into account. But as no test is perfect, we always look at mitigations. Inez, do you have something to add?
Sure. Yeah, to give you an idea, our current class 3 products, the performance for sensitivity is about 98%, and specificity is 99%. So the tests are specific and sensitive as well. So that to give you an idea of the likelihood of getting a false positive or a false negative result there, we try to minimize that with these class 3 devices.
And it looks like Uwe has come on camera. Uwe, would you like to add?
Yeah, Uwe served just one additional point to Inez's communication. I would like to emphasize that the down classification does not imply that we are expecting a difference in performance. So what Inez just mentioned is we have these high-performing devices that are currently brought to market via PMA, market approval, but we are not expecting that a 510K approach would then be coming to a scenario where the performance is lowered.
Exactly right. Ms. Schwartzkopf, did that answer your questions? You're muted, but your hand is still up. So I want to make sure we were.
Yes, it does. Thank you.
All right. Dr. Pereira, and I know I keep messing your name up.
Oh, that's okay. Marcus Pereira here from Columbia University. So I think the main question I think that many of us have had it, I think it revolves around sort of these standards in terms of false positive and false negatives, and sort of what are some of the minimum criteria for approval for test performance regarding class 3 and class 2. It sounds like I'm interpreting this that there's no minimum standard, but also no difference in expectations or requirements between, at least in terms of test performance, between class 3 and class 2. At least that's how I'm interpreting things. Is that correct then?
So when we write the special controls for something that can be down classified, we can put those performance expectations into those special controls. Uwe, I'm sorry, I may have interrupted you. Did you want to add anything?
Oh, no, I think Uwe Schurf again, Tim, no, this is exactly what I also wanted to share, that the opportunities for us at FDA to capture necessary kind of informational pieces in the special controls, and they can be related very specifically to performance. And that's something that I think normally is not known in the diagnostic community because we're not doing this every day, but we have, again, that tool in our hands, and we have used it several times.
That's
special.
good.
And
I'll just clarify that for everybody who's attending today, if they don't know what special controls are. This is a document that would be generated when the down classification process goes through in order to translate the expectations into written form for developers. The FDA then uses those special controls as a guide to determine whether a 510(k) submission is sufficient for authorization. Thank you.
And I will remind the panel that question number 2, because you're not seeing them all in front of you, I do have my paper in front of me, but question 2 is actually going to get onto this topic exactly, which is how to mitigate those risks. So I want to stick with question 1 just to make sure that I've heard everyone and we've all had a chance to speak about, do you think this list of risks is comprehensive? Is there anything on here you think shouldn't be on here, or is there anything you want to add?
I wanted to make sure, is vaccines, I think it was mentioned in the summary, but I'm not sure if it was mentioned in the slides. So regarding for hepatitis B in particular, and the sort of test performance, whether a false negative or false positive particular surface antibody result would lead to either unnecessary vaccination or not vaccinating someone when they would be eligible for the vaccine. I think that was mentioned, but I want to make sure that that's included.
Okay. I'll take a note of that, and I'll go next to Angie. You put your hand down. Did you still have a question? Or has it been answered?
No, I just wanted to make the point about there's no reason that the sensitivity or specificity requirements would change just because it was down classified, and that's already been done.
Okay. Dr. Bloomberg?
Hi, Emily Blumberg, University of Pennsylvania. It seems to me from hearing from the public comment that the main concern from the public is access to care. And access, so that leaving these as class 3, is there a greater risk that fewer people will be tested? And I'm not sure I think that's probably related primarily to the cost of bringing the test online. So the one thing I would add is whether leaving them as class 3 will limit access to these tests.
I think I would reemphasize what some of the panel members said, is that there are many tests that are available ex-US that people haven't tried to get through. The PMA process, it's not just the cost, but there's a lot associated with it, including the post-marketing efforts that one has to go to keep that approval in place. And I think that, so it's not just about who can afford to do what, but it's whether or not we really want to encourage more market competition by making it somewhat easier to get through the process, because that helps public health overall. But the risks, I don't think, are affected by that. So sticking with that risk conversation. Dr. Moore.
Yeah, thank you. So my question, and I don't know how whether this is something that could be provided here today. I know that CBER is in charge of the HIV testing, but I know some of these other discussions, same discussions have been had with HIV testing going from level 3 to level 2. And I wondered if there was somebody who were involved in some of those discussions who could help inform our debate today. Provide some background.
Is there someone with the FDA who was involved with any of those changes in classification?
This is Tim Stenzel. And Uwe, do you want to add anything? I would just state that we take this risk-based examination on an analyte by analyte. So what was done for HIV may not be completely overlapping and appropriate necessarily for HBV. We do know that other than OTC, diagnostic devices for HIV were down classified. So we know that result. But Inez or Uva, anything to add?
Yes, I would echo what Tim said. We do take into consideration, everything that is discussed in the panel for deciding whether the reclassification process should proceed for HBV, as well as in putting together those special controls. We also take into consideration the whole HBV environment, the need for the availability of specific tests, the therapies that are available, medical practice. We have discussions with other experts to put all of this in development.
Thank you. I know it's apples and oranges, but the thought process is similar, so I wasn't sure if thank you for that.
Yeah, I appreciate the discussion. This is Tim Stenzel. I would just add that we've already down-classified HCV, which is maybe more closely aligned with HBV than HIV.
Dr. Lahoz?
So if I understand correctly, we have a fair amount of information regarding the performance of hepatitis B testing, currently classified as 3. And the diagnostic performance being quoted, 98 sensitivity, 99 specificity. And on the other hand, we have a risk to the population, those that may have a lack of access to care, where if they were more availability for testing, they could be diagnosed promptly, engaged in care earlier, and then subsequently prevent some of the long-term complications of hepatitis B, which are liver damage, hepatocellular carcinoma, and liver failure, as it's been quoted. So I think that to me, the risks have been outlined, and I can currently not think of a additional risk to add. And it appears that if we are going to put adequate performance standards, we're going to have post-marketing surveillance to ensure that the IVDs continue to perform at the same standard, and it's going to increase the availability of testing. It appears that the pros of potentially downgrading will be larger than the cons.
Thank you. Dr. Caliendo.
Angie Caliendo, Brown. I want to go back to what Emily said, and Barbara, I may not have understood your response, but I do think keeping it as a class 3, the risk is reducing access to care. And is that not something we would add under the risks of? I mean, I might have misunderstood when you said that really wasn't
well, it may be a benefit. I mean, it depends on how you look at things, right?
Well, since we're not listing benefits, I think.
Well, we will. You're right. We're not. Okay. And so, my personal bias is any test not done is a test that's failed. So restricting access is a huge risk to the population as a whole. Right.
And so that may be a risk we need to add. Yeah.
So okay, I'll put that in my list. The risk to public health of limited access.
Camille Cotton, Massachusetts General Hospital. I just wanted to bring up.
Can I pause you for a second because? Oh yes, you are next. I'm sorry. I thought Dr. Bradford was next, but you are next. So sorry. Let me uninterrupt you.
I wouldn't do cuts on Zoom. Sorry. So Camille Cotton, Mass General. In general, I am in favor of downgrading. I think that there are a lot of advantages. One of the issues I wanted to highlight that's something that I find really occasionally upsetting clinically is when we give a hepatitis B vaccine, we can actually detect vaccinemia for up to 2 weeks or so after they receive a vaccine. So I could envision someone, for some reason, getting hepatitis B vaccine and then maybe thinking a lot about hepatitis B and getting an point of care test that might suggest that they have positive hepatitis B surface antigen, which in my clinical world is always kind of a dramatic diagnosis, especially when they've been negative, and then all of a sudden are positive because we think that's real hepatitis B, but it turns out it's just vaccinemia. So somewhere in the package insert or elsewhere, I would want this issue.
Could I ask you to hold this, and the next question is about mitigations, and that's exactly where this should go. So if I call on you first when we come to the next question, and let me see if Brad, did you have anything else that you wanted to add on risks? And then I'll summarize risks, because it's clear we all want to talk about mitigation.
Yeah, that's why I've been raising and lowering my hand. But Brad Spring with Roche Diagnostics. Point of clarification for FDA, because I think there's a future question on here too around expanding or changing intended uses. So in light of looking at the risks associated with these tests and down classification, is the assumption is that the intended use is not changing? And I'm assuming yes, but you can answer in the affirmative or negative. But I think looking ahead, some of these tests may see a change in their intended use. And should we be considering that when we also think of down classification and risk associated? I know you can't think of them all, just because it may be somewhat infinite, the possibilities, but just want to see if there's any clarification you can provide on some of the decisions we're making today based on that statement. Yeah, Tim. All right.
Yeah, I'll start, Tim Stenzel, and then Inez has come up. So it could be that sample types change, settings change, so that would change intended use statements. So those are potential risk mitigation opportunities for sure, and we certainly take that into account as we assess a new test. And would want to put as much as we can into the special controls. And as it looks like you've come up, you may have something additional to add.
Sure. Yeah. So our intended uses are adaptable. They're dependent a lot on medical practice or anticipated changes to medical practice. We also develop the intended use in conjunction with how the clinical study is performed. So a big part of the intended use is its support by the clinical study. So there is flexibility there.
Yeah, okay. No, that's good. It was just around the impact of a new intended use affecting the classification decision. And maybe this is something we can hold to the last question.
Dr. Stenzel.
Yeah, I just wanted to add, there was some discussion about whether you all can discuss benefits, and the FDA team would really appreciate benefit discussion in this when we get to at any point that Dr. Vanderpool you deem it appropriate today, and not just for this analyte, for all the analytes. Thank you.
I think that's really critical. I'm going to try and summarize so that we can go on to the mitigations, because I think that's one of the places where everybody has a lot of input that's at the tip of their tongues. So Dr. Stenzel, with regard to question 1, I think the panel generally believes that the risks are predominantly inclusive and exhaustive. So the 2 things that the panel had concerns about was that some of those risks needed to be quantified. And I'm going to paraphrase, but one of the things that I heard was that those risks may be use case dependent. And so, for example, for people who have just been vaccinated, the risk is different, obviously. And for very low prevalence populations, obviously, your positive predictive value changes. So the sensitivity and specificity have different impacts, even though they don't change. They have different impacts in different populations. So I think that while the risks that you've covered are comprehensive, they might need to be sort of restructured in the thinking to provide a little bit more detail for people. And then I think that given the information that we had about the performance of current class 3 tests, it sounds like those risks have been in part addressed by having really high quality assays. And that would move us on to question 2. So if we could put question 2 up on the screen just to remind everybody, this is the point at which we should discuss the mitigation measures and controls. And so mitigation measures can be describing what populations it's appropriate to use these or what clinical context it's appropriate to use these. Controls can also include setting a minimum sensitivity or minimum specificity that the assay would have to hit. So it's not related to class 3 that they have to have high sensitivity specificity. Class 2 assays can be required to have the same performance levels. And I think that this is the point at which we should also discuss benefits, because the benefits obviously might not mitigate the risks, but maybe outweigh the risks to an extent that we think that it's worth talking about those at this point. I'd like to start with Dr. Cotton since I interrupted her twice already.
Thanks. So I just wanted to bring up the issue of hepatitis B surface antigen positivity within 1 to 2 weeks after receiving vaccine, which happens, I would say in my institution, happens several times a year. And if this were outside of a standard medical context, there might be concern about the diagnosis of acute hepatitis B, in that it sure does seem to look like it. Although there, with this point of care testing, there wouldn't necessarily be LFTs in clinical interpretation. So I just wanted to highlight that this is a potential major issue that would seem like it should be covered in the package insert or other consumer-based education or other areas that the FDA and others are important, but just an area where I think we should consider risk mitigation. It's beyond my area of expertise, but I don't believe that altering thresholds for positivity or things like that would really be able to handle this problem. But happy to hear what others might have to say. Thank
you. You're probably right about that. And I think that I'm going to remind the group once again that down regulating from class 3 to class 2 does not mean we're talking only about point of care tests. These might still be laboratory tests. And so it may be that point of care tests need even different special controls. So, I mean, and that's for the agency to work through their process. I think I'll go on to Dr. Prokop.
Hi, thanks so much, Gary Prokop, American Board of Pathology. Just a couple of points. One was we talked earlier about it doesn't necessarily mean that the performance standard can be lowered. I think when we're entering these conversations about whether we should reclassify, it would be great to open with lower performance standards will not be accepted. You know, the performance standards will have to be as good as they were at class 3. The other point is I really did appreciate the point earlier about differences in special controls with respect to point of care testing versus laboratory testing, et cetera, because we do know there are some differences in reliability depending on who's actually doing those tests. One of the things that would be helpful, and I know this might be getting into the weeds too early, but when we consider some of these specific tests, it would be useful to know what the specific special controls might be that would ensure that quality. Just some thoughts. Thank you.
One of the special control, I don't know if it's a special control, and I'd like the FDA's opinion, but one of the issues is obviously that it might never receive a CLIA waiver. So it still might require laboratory trained technologists to run the test, even if it's at the point of care. Because remember, a point of care doesn't mean that an untrained person can necessarily run it. So Dr. Stenzel, did you have thoughts you wanted to add about that?
Yeah, so we can write special controls and I should say that that there are some analytes. First of all, the presumed or the baseline classification for any new analyte that the FDA hasn't seen before is a class 3. We can, when we interact with a test developer, decide even from the very beginning that we have enough knowledge to write special controls to justify a de novo pathway, which will lead from the baseline state of a class 3 to class 2 with special controls. And when we write those special controls, whether we're in the process of down classifying that we might do for any of the analytes today, or when we originally on the first developer interactions, we decide that it is okay to be a class 2 device, we can write special controls for all different situations, whether it's central lab, point of care, and or over the counter. We can write them if we feel like we can have one regulation and put it all into the special controls. We can do that if we feel like we have enough knowledge about the various risks and mitigations to justify that. And then the special controls will take that into account, whether the setting is central lab, point of care, and or OTC, among different sample types too, and different clinical uses. Thank you.
Thank you.
Thank you. Dr. Beavis.
Thank you. Kathleen Beavis from the University of Chicago. For me, the largest risk, and this follows up on what Dr. Cotton mentioned earlier, but for me, the largest risk is of a false positive serologic assay that can lead to unnecessary lifelong therapy. And I've got to wonder, could this be mitigated by specifying that, a single serologic assay alone should not be used as the sole basis for instituting therapy, whether we ask for repeated serologic testing or, my preference, viral load testing before the institution of therapy. So that would be my suggestion to mitigate that risk.
Sorry, can I ask you directly what the current clinical recommendation is? Because even with a class 3 device, isn't there some recommendation for repeat testing, or is it really, as you described, a single test and then therapy?
Is there a recommended testing algorithm?
There could be. I just know that our Kathleen Beavis, University of Chicago. I just know that our practice is to follow up with a viral load.
Does anybody recall whether CDC has an algorithm? Dr. Petty, you're raising your finger, so maybe I'll call on you and see if you have information about that.
I'll let Dr. Moore first, and then I'll follow, since we're the clinicians here.
Sorry to jump in. No, there are
Don't forget to introduce yourself, please.
Sorry, this is Dr. Moore, University of Kansas, or Wichita, Kansas. So there are algorithms that are published by the societies, and the hepatologists, the gastroenterologists, infectious disease physicians, we all tend to follow those guidelines. I'm unaware of any clinicians that are prescribing lifelong antivirals for hepatitis B without the input of these specialists like us. But certainly that's my experience in my community. CDC, I'm unaware of any specific CDC guideline, specific CDC protocols or algorithms with regard to this, but there are recommendations that are made. Anyway, that's all. Thank you.
Thank you. Dr. Petty.
Yes, and Dr. Beavis, I completely agree.
And don't forget to introduce yourself so the transcription.
Yes. Hi, Kathy Petty, HealthSpring Global. Dr. Beavis, I completely agree with you that a single test does put high risk because the test results have extremely high consequences. And I would like to reiterate, while we think about declassifying, down classifying this to class 2, the particular wording and product labeling as an aid in the diagnosis, I feel very strongly verbiage like that should remain when we consider class 2 device. As a clinician as well, we use a constellation of various different laboratory tests, including ALT, when we decide whether or not to treat or observe. Secondly, in the analytical validation, when we're talking about special controls, I know a lot of us in the laboratory industry feel pretty strongly that rigorous validation requires a pretty high N. And if we can maybe set thresholds or guidelines on the tests needed to be run to meet these sensitivity and specificity thresholds, agree with Dr. Prokop. I don't think that's negotiable, needs to remain very, very high. But at what N? And I think we've all seen that the more we test, it's a much more rigorous analytical validation. And finally, I don't want us to lose sight of the fact that hepatitis B has international standards. So we're talking about an analyte that does have international standards, which makes, particularly among manufacturers, test performance being more harmonized. And for clinicians to be able to rely on these test results that is interoperable or portable across various different testing institutions. Thanks.
Thanks, Dr. Petty. Brad?
Hi, Brad Spring with Roche Diagnostics. A follow on to Dr. Petty's comments, I more or less want to make a statement and an ask. I've been around a while with diagnostics and seen many down classifications, and I don't recall any that have been down classified where the performance standard has been lowered. The only time I've seen it change is in an up classification that was with the flu tests that many of you may have remembered that the performance did need to meet a higher threshold. So completely agree that the performance threshold should not change. But Dr. Petty mentioned this too. I think some of the barriers of market entry are around the levels of evidence needed to establish that level of performance. And while I'm not necessarily asking for significant reduction in the amount of testing, it's really around considering alternative types of evidence when we look at testing, because prospective trials are probably the most burdensome approach to establishing evidence, mostly because of finding the right type of patient and the right type of specimen. So I would encourage the agency, as we look at this approach of special controls, to look at real world evidence standards, which I think is something we've been talking about a lot in the industry. Are there specimen banks we can leverage that are trusted? Are there other types of evidence we can use to establish that type of performance standards? Because I think that is one of the main barriers. And I think Dr. Vanderpool, you mentioned this too. One of the biggest changes we would see with a down classification would be the reduction in the post-market pre-approval prior to implementation of any sort of change. But I would add, from an industry standpoint, that does not mean we change the testing that we do to then make a modification. We just would not require for certain types of modifications a pre-market clearance to do that.
Thanks. Dr. Caliendo?
Angie Caliendo, Brown. So I just want to reiterate the performance characteristics shouldn't change. Kathy made a very important point about used in conjunction with. Several people have said that. The other thing that I was thinking might be helpful, since the goal here is to increase the number of tests that are available, increase access. So maybe instead of comparing it to one predicate, comparing it to two predicates when you're doing your studies, I'm envisioning a lot of different groups coming into this. And even though there is an international standard, is that a way to mitigate variability from one commercial product to another? So just put that out as a potential special control.
Thank you. Dr. Bloomberg.
Emily Bloomberg, University of Pennsylvania. One other area that I think needs to be addressed in terms of mitigation is, it's my understanding that public health reporting goes through the labs that perform the test, not through the clinicians. And so you need to make sure, if we're talking about things like, for example, what Dr. Cotton was talking about with these false positive surface antigens, that there needs to be clear standards for public health reporting to make sure it's accurate for both good and, for both characterizing disease and also avoiding unnecessary stigma for patients.
Agreed. Dr. Chu.
Hi, yeah, Charles Chu, UC San Francisco. I just wanted to make a point about the potential difference between qualitative and quantitative assays, because we're really considering a panel of 4 different assays. And in my mind, serologic testing for, say, surface antibody is quite different than HBV DNA. I mean, those are tests that are used for widely different purposes. You would never order an HBV DNA, for instance, for diagnosing as a primary diagnostic test for hepatitis B. So there's certainly a risk, especially with the quantitative assays, of inappropriate use. If these are going to be used for screening, for instance, there's a potential risk that they're not being used appropriately. In addition, there's a much more challenge with regards to standardization, because these are quantitative assays and, based on, say, interpretation of viral loads or response to therapy and viral loads, different tests can have widely different performance characteristics. They may not be linear. They may have different limits of detection. And so with regards to the molecular test, which is the HBV DNA, I'm a little concerned about perhaps declassifying that. And the question that I want to ask the FDA is, are we considering these tests as a whole, as an aggregate, or are we considering each test independently?
Dr. Stenzel? So when we sum up, the question number 3 is going to be sort of your recommendations, the panel's recommendations. And I think the question for you just now is, do you want those recommendations by assay or as in aggregate as a group for HBV tests?
If there's specifics per assay, then I would provide them to us if there's specific concern. So, we were, and I think there absolutely are differences between serology, antigen, and molecular tests, and there are different roles and purposes for diagnosis.
Okay. Mr. Spring and Dr. Chu, if you're finished, if you'll lower your hands, or if you have a continue on question, leave them up. And Dr. Karsner, you are next.
Hi, yes, thank you. My name is Ryan Karsner. I'm the Deputy Assistant Director for Hepatitis and General Viral Infections branch in the Division of Microbiology Devices. I'm also a internist and infectious disease physician. I just had a couple of points of clarification. One is, as we discussed, the intended use statements for all of these assays are as an aid in the diagnosis of hepatitis B. And we also have language in the labeling of these tests that the test results should be interpreted in the context of, the clinical picture and other test results. And then the last point of clarification is that the CDC does have recommendations for interpretation of hepatitis B serologic test results. If you search for that, it'll be the first thing that comes up, and it'll tell you, it has a table with hepatitis B surface antigen, total anti-hepatitis B core, and anti-hepatitis B surface antigen antibody, and how to interpret those results. So there are recommendations that are published.
Would have been shocked if not. Dr. Garcia.
Hi, sure. Ines Garcia. I wanted to touch on Brad's point that there are alternatives to the clinical study. I mean, we are considering real world evidence. We also do consider retrospective samples as a way to supplement. Of course, where it gets very tricky is when you're talking about like a CLIA-waive type test with finger stick, which is a sample type that is much harder to store.
Very true. Dr. Caliendo.
Angie Caliendo Brown. I just want to make a comment in response to Dr. Chu's concerns about the quantitative DNA assays. Yes, they're quantitative and they're more complex, but also the requirements are different. There's going to be an LOD requirement, an LOQ, there'll be linearity, there'll be reproducibility. And so what the FDA will require of a quantitative assay is different than what they'll require of a qualitative assay. And I think that as long as those performance characteristics are what they are now, we have a lot of experience with these assays, we know they work, that down classifying them doesn't necessarily increase, in my opinion, doesn't increase the risk as long as we stick with those characteristics. And I think, you know, there are differences in LOD, there are differences in LOQ, and that's one of the reasons I said maybe more than one predicate device, right? So you can get an idea of how these compare across more than one test. But for me personally, that being a quantitative assay to me doesn't make me uncomfortable with down classification, because I think the FDA has a lot of performance characteristics that are required to assure how it functions.
Great. Dr. Perea?
Yes, Marcus Pereira from Columbia University. I think 2 points, just also in response to Dr. Chu. I mean, the quantitative surface antibody test is often used for us to determine whether someone has responded to the vaccine or not, based on certain thresholds. So that hopefully that will also obviously stay the same in terms of its sort of test performance and quality. So that's one minor point. The other sort of thought I had in terms of risk mitigation is, you know, when you sometimes order the panel of hepatitis B testing, the surface antibody, surface antigen, and core antibody, embedded in the results, there is an interpretation, because obviously these are complex tests, and understanding those, as one of the slides during the initial presentation included, sort of you can have different interpretations about acute, chronic, you know, isolated core antibody positives and so forth. So hopefully the downgrade wouldn't remove that sort of helpful interpretation. And I'm thinking more not necessarily for specialists or those who use those tests often. And I think we generally have a facility in interpreting those. But if the idea is to increase testing and more providers who may or may not be that familiar with interpreting those tests are ordering them, it would be important to have some way or some helpful tool to help them interpret those complex tests.
Okay. Dr. Stenzel?
Yeah, I just want to reiterate that the standards we have for current class 3 tests don't have to change necessarily for the down classification to class 2. Standards can be the same. What we're really talking about is the amount of work required for the authorization. The time to authorization, a 510(k) submission takes 90 FDA days, and PMA much longer, and the costs of the submission. All those do impact potentially the number of developers who invest in developing a test. But again, the standards, you know, you can make the recommendation if you want that the standards don't change going from class 2 to class 3. Thank you.
Thank you. Dr. LaHose?
I am reassured by both the comments of Dr. Ryan Karsner and Tim Stenzel that the, you know, comments, stipulations surrounding how to diagnose hepatitis B would not change with the potential reclassification. But maybe a comment on what Dr. Caliendo and Dr. Chu have mentioned before about reclassifying all of them at the same time versus some of them, is the possibility that those populations that don't have access to care may have all of the tests done performed at the same time, just to adequately categorize them as only core positive versus core plus antigen plus DNA plus LFT abnormalities plus abnormal INR. So if we were to just reclassify a few of them and not all of them, we may end up with, again, a potential disadvantage to quickly diagnose them and you know, assign them a certain degree of severity or priority to treatment.
Thank you. Dr. Chu, back to you.
Yeah, I think Dr. LaHose expressed it really well. This is my main concern. My main concern is that if we down classify all of these at the same time, that in my mind, if the reason for down classifying in the first place, if the primary reason is really to improve access to care, you would only really need to down classify the serologic tests. And then, the quantitative serologic tests and the quantitative DNA tests, those are used once a patient's in the system. You want a patient being followed carefully by an expert, if a patient really has newly diagnosed hepatitis. And so why can't we selectively down classify, for instance, the serologic test and ensure that patients at least have screening and have access to care, and then get them into the system where you would send off all of these additional tests, because there are more tests than just these hepatitis tests, as you remarked. You have to test INR, liver function, et cetera, and you want to if your purposes of it is really for screening, then why do we need to down classify all of them at the same time? I think that's my question.
And I think I'm just going to reiterate that the purpose isn't just for screening. The purpose is to determine whether or not we have enough information to say that special controls are sufficient to get these products to market. And I would also, just from an STI perspective, there are now molecular tests that are available at the point of care. They're not all CLIA waived, so some still need laboratorians to run them. But I think that one of the things when people are talking about access to care is we have to remember not every person in the US lives in an urban center where they can go in. So just because you have follow-up tests at large urban healthcare centers doesn't mean that people all over the country do. And we are a leader in the world. And I think this has global impact. So I would just throw those things out for your consideration. But I promise you, when we get to the next question, I will call out 5 different types of assays so that everybody has a chance to be heard again on that topic. Dr. Petty?
Yes, Kathy Petty, HealthSpring Global. I just will reiterate what you just said, Dr. Vanderpool, that I think bringing it down to a class 2, the added benefit is increased access. But I strongly believe that we've had years of experience and the technology has proven itself to be reproducible, accurate, and reliable. And for those reasons, we are data informed that class 2 would allow us to deploy these tests in a safe and effective manner.
Nice summary. Thank you. Dr. Caliendo?
And I would just add that one of the points that the FDA made in our introduction on classification was that tests should be classified at the lowest level possible based on safety and efficacy. And so that's part of their mission too, is right. What is the safest based on safety and efficacy? What is the lowest level where they can, with special controls, down classify?
Dr. Winston.
Yeah, I think there is some perception and probably experience among many of us that we have like this. We have the PMA process, which is like extensive, elaborate, and can be a barrier, but assures that the tests are properly evaluated. And then we have the class 2, like 510K process, which sometimes is viewed as, okay, very minimal standards and maybe concerning. But maybe can we hear some examples of special controls, like how you can move between these two extremes and how you can make sure that the standards are truly not changing? Because I mean, there is a concern. I mean, there is a difference in the process, and that concern is probably what makes some of us hesitate a little more, and while wanting to lower the barrier, not risking any deterioration in test performance. So I think that it would be great to hear a little bit more what could be done and how we could move like somewhere in the middle ground.
I can start, and then if any of my FDA colleagues want to add in. So again, I'll just reiterate that the standards you can make the recommendations that the standards for each type of hepatitis B device stay the same or equivalent to what is right now. And we can certainly move forward with that if that's the best thing to move forward with now, and see how that works. There may be other mitigations, though, that allow, say, easier development of over-the-counter or point-of-care tests. And when you look at the benefit risks, it could be that a slight decrease in sensitivity or specificity is outweighed by the fact that there is a lot more access to testing. I think there's, as we saw with the pandemic, access to testing at home was really important to help the spread of disease. And that's really what we're trying to get at here is can we eliminate the spread of hepatitis B? Can we limit and mitigate the morbidity and mortality from hepatitis B? Can we make diagnoses more early in infection? Can we prevent vertical transmission? All these things are huge benefits. And when you look at benefit risk calculations, and we can model this and are open to modeling this when you have measured performance differences, though slight, between, say, a point of care or over-the-counter test and the increased access to testing. We can also mitigate by, hopefully, it says my internet is unstable, so hopefully you can still hear me. We can mitigate through labeling. So this might be, you need to go see your clinician and you need repeat testing, things like that. So we can special controls aren't just writing in necessarily exactly what's, say, in a PMA product right now, but we can add, for certain situations, we can add additional mitigations through the down classification process. I see my colleagues Inez and Uba might have something to add as well. Thank you.
Hi, Ines Garcia. Yes, I wanted to build on that. So the special controls do have some built-in flexibilities in case there are alternate approaches that come up. They are generally written to describe the types of studies that we would expect to see, for example, like cross-reactivity, precision, the analytical studies. In general, they may describe a clinical study or method comparison study. To give you an idea, also there could be labeling mitigations, such as limitations that need to be added, and other special considerations with specific environments in which the testing should be performed, or any kind of instructions for the training of the test operators. So to give you an idea of what some of the special controls may look like.
So can I just, to clarify, you could, in theory, you could have a special control that asks for a PMA-like trial or trial data but it's still like a class 2 approach. Is that correct?
Exactly. You got it right.
Dr. Scherf?
Yeah, Uwe Scherf, FDA. Just to add to Tim's and Inez's description here. Yeah, it's correct. So and the point here for HBV is also something I think that we would like to share. I mean, we have been reviewing these products for 20 years. They have been out in the market, and I think it has been mentioned earlier that they have established their performance and their use in the market. When we now think about down classifying, it doesn't mean that, first of all, we need to remove or decrease the performance. We do not need to remove any evaluation approaches like WHO standards. I mean, we will not do that because this gives us, even in the PMA environment, the assurance it's safe and effective. And there is no need for us to kind of move this or move away from that because it's part of the thorough evaluation that will also happen in the 510K paradigm. Now, it was mentioned by our NCI colleague that 510Ks are maybe less strictly evaluated, I would not necessarily stand on that word. Yeah, they are differently evaluated, but to a level that at the end of the day, FDA and our medical colleagues can determine they are safe and effective, and they are willing to use them.
Dr. Pereira?
Yeah, Marcus Pereira of Columbia University. I'm going to bring up something that I apologize to my colleagues. I don't have my thoughts fully together, but I guess a question about reflex testing, which we do have for confirmatory testing, for example, for the diagnosis of HIV, and sometimes when there is a sort of indeterminate result or even sort of as a standard. And we don't have that in hepatitis B. And obviously, it's a much more complex situation with different sort of states of infection or non-infection. But is that something that, in the appropriate situation, which would have to sort of fully outline, is that something of a possible special control in certain circumstances?
Tim Stenzel, FDA, yes. We could put that in as a special control for new settings of testing, for example.
Dr. Lajos.
This is Ricardo Laos from UT Southwestern. I think that what I am hearing from our colleagues from FDA is that the hepatitis B tests have been evaluated for over a decade with good diagnostic performance. We've also heard that there's a fair amount of safeguards to ensure the performance of the tests, if they are reclassified, will remain the same with a high sensitivity and a high specificity. Also heard that we can keep the same statements regarding the complexity of diagnosing hepatitis B with regards to putting the test results in the context of many other things that I assume that they're well stipulated in the class 3 classification. So to me, if we have all these safeguards, the predominant pro is really increasing diagnosis at multiple levels, which is preventing vertical transmission, preventing transmissions amongst populations that currently have a high prevalence of the disease. So to me, I'm hearing that there's a fair amount of safeguards in place, and the big pro is improving access overall. One comment that was made was, which information will be provided for those seeking approval for a class 2. I think if we make the bar so high that it is similar to a PMA without necessarily being a PMA, we may defeat the whole purpose of what we are trying to achieve. So the safeguards need to be there, no doubt, but the safeguards need to be enough that they provide us with the reassurance that they will perform correctly, avoiding the risk, but not high enough that, again, it eliminates the increase in access.
That's some great points there. I mean, I think that gets us kind of into considering what is the absolute minimum sensitivity and specificity. If you're going to put special controls in place, what do those need to look like? And we've heard from several people that they have to be exactly where we are right now, which is 98, 99. Is that what we really need to provide safe and effective, good quality tests? Again, recognizing that every test not done is a failed test. So, maybe those special controls need to be considered, but I don't think that's really the purview of this panel. So I'll move this on and let Dr. Ng get in her 2 cents worth.
Thank you. Valerie Ng, Alameda Health System. I wanted to bring up the issue of harmonization. If we move it from just a few manufacturers today to many more, I do know my current assays, the IFU clearly states you cannot mix and match results from different manufacturers. So then that gets to specifically issues like the qualitative and the quantitative surface antibody test. Are we going to peg the interpretation at 10 or at 12, whatever the units of measure? Are we going to have one being more analytically sensitive than the other? And then how do we interpret that? And then I want to talk about the surface antigen. My current assay involves an initial positive must be tested in duplicate and then must undergo a neutralization. How are we going to assure other formulations of surface antigen tests have the exact confirmation so we are not managing a series of false positives, which is why I think I have this whole reflex testing program for that particular test.
And we're going to come to that with question 4. Because that's a valid point. Dr. Petty.
Kathy Petty, HealthSpring Global. I just wanted to clarify an earlier comment I had made about special controls and the sample size calculation and power calculations in the N. The N I meant was demographics. I know the FDA does this, but would like to emphasize again that we have the appropriate patient populations represented, the vaccinated, the unvaccinated, those at high risk and those at lower risk. I think that would be very, very impactful in better understanding test performance.
Agreed. Dr. Stencil.
Yeah, I'll first respond to Dr. Petty. Yes, we like a diversity of the patient population, and we have a new diversity enhancement policy that we really want to include patients from all different groups as well. I also wanted to respond to an earlier comment, and I forget who it was. It might have been Dr. Winstonson, about if we make the bar for performance as high, no, I think it was Dr. Lahaz, as high as it is for PMA, and then you're going to try to get point of care or over-the-counter tests that could be incredibly valuable. It can definitely act as a disincentive to develop. If we have potentially seen this with hepatitis C, even though we have down-classified it. We have not seen a tremendous upswing in submissions, and we essentially matched, in the special controls for the now classification of hepatitis C, the performance expectations that we saw in PMAs. So, and we do have some feedback that may be inhibiting innovation here and the ability to access testing for hepatitis C. So that is something certainly to consider. The panel should consider here is ultimately, what is the goal in hepatitis B testing? How can classifications of the device impact reaching the public health goals of hepatitis B testing? Thank you.
Dr. Winston.
I fully agree with your statement, and but I think it goes back to the fact that there are different indications and different types of tests, and I think for like a point of care test that can extend coverage of the test, like accepting a slightly lower performance would be a great trade-off, but for other tests that we're discussing, we may not want that. So I think that I think that it sounds like that is achievable through a class 2 process through the special controls. And I think that is, to me, that is a really very reassuring message to hear about moving in that direction.
And I think that's one of the areas where mitigation via labeling helps us, right? So we can actually say this test has somewhat lower sensitivity, so you must do a confirmatory test or some sort of reflex test, which we've discussed. Dr. Prokop.
Well, you just stole what I was going to say.
Sorry.
So, I get the idea of, potentially lowering, say, sensitivity, specificity in order to increase access. I will also say erroneous test results cause harm. So that's something to consider too. So we don't want, we're going to really have to walk that tightrope. And I think that tightrope, it really is mitigated. If you lower, you have to impose a diagnostic algorithm that will right the wrong.
Mm-hmm. Couldn't agree more. Has everyone gotten everything off their chest that they wanted to share, or I'll try to summarize unless I see another hand or two. Dr. Moore?
Just one thing, I see, basically spend all day seeing patients, and I get a lot of calls on about false positive Hep B core IgM testing, which is problematic. But I was just going to say that just from my perspective that I think if we have increased access to care by making inviting product developers to bring assays to the market by downgrading it to level 2. That's, from my perspective, I think that would be the best option. But leaving that would be the qualitative testing and the serologic testing. But the quantitative testing, either bringing it down to level 2 and having a special control on it, or leaving it at level 3, that to me is the most critical aspect, because we really don't want to have false negative. I know that downgrading to level 2 doesn't imply a reduction in specificity or sensitivity, but there is that possibility that may happen. I think we need to leave that at level 3 or level 2, very significant special control.
Okay, thank you. Dr. Stenzel.
Yeah, and thank you, Dr. Moore and Tim Stenzel. Yeah, definitely we can have more or less flexibility around different technologies as we don't classify. And it may be that those technologies that are largely used to confirm or provide additional information after diagnosis or confirmation of diagnosis can remain at a higher bar than something that would allow point of care or over-the-counter testing. Thank you.
And this is a place probably at which we could consider, different controls for screening versus diagnostics, right? We don't always disentangle those, but we often should. And I think maybe this is one of the places where that would be appropriate. Okay, so I'm going to sum this up. What I'm hearing is that in general, the panel believes that there are mitigations that should be put in place. Those mitigations include labeling about when it's appropriate to use. Maybe we say these tests aren't appropriate to use within a month of vaccination, for example. But labeling mitigations, intended use mitigations that cover what clinics or what circumstances testing is being done, maybe disentangling diagnostics from screening. Requiring follow-up testing was something that we heard fairly often, or confirmatory testing, and that labeling must include language such as an aid in diagnosing, again, suggesting that further testing may be necessary, but also that the interpretation has to be embedded within the clinical context. And so then we've talked about also those special controls, including the sensitivity and specificity to either remain where they are with the assays that we currently have available that originally achieved approval using the class 3 process, whether we need to stay at those same levels of sensitivity and specificity, but regardless, there do need to be very carefully considered special controls there, and that qualitative and quantitative assays may actually have different needs in their labeling and mitigations. We also had concerns, as I just mentioned, to make sure that we differentiate between some of these different needs, and that it's not one blanket set of mitigations, but it may have to be assay specific or type of assay or area of use of assay, so point of care versus laboratory-based testing. So Dr. Stenzel, is this adequate, or did you have further questions on the topic of mitigation? You're muted still.
Apologies, Tim Stenzel. Yeah, I'm satisfied, but I want to give my colleagues at the FDA who are on the line. It looks like no one else has anything to add. So thank you so much for this.
Yes, it was a really robust discussion, which I think is very helpful. And I appreciate the different perspectives that everybody's bringing to the table. So let's go on to question 3. And I will remind you, so this conversation is based on the information presented here. Discuss whether or not, based on the available information, the panel believes the FDA should initiate the reclassification process from Class 3 to Class 2. Again, I will remind you that we can contextualize that by different types of assays. So antibody assays, antigen assays, molecular assays, qualitative and quantitative, however the panel feels that that's appropriate. I would also remind you of the information that we were given in the very first presentation from the FDA, that we are really supposed to help the FDA make the decision about whether or not there's enough information available and enough data available to inform creating special controls, because if there is that level of information, then it would be appropriate to move to Class 2. That many things come at Class 3 while that information is lacking. So with that in the back of our minds, I'll open it up for conversation about whether the panel believes that the FDA should start that process of down classifying HBV test. And you can put that in terms of whatever assay you want to focus on. Dr. Bloomberg.
I'm going to vote or speak in favor of.
I'm going to just remind you that we're not voting.
Oh, sorry.
So
Yes.
just to make sure that we're clear and we don't cross any boundaries that we're not supposed to cross.
Thank you.
That said, I'm sorry, please go ahead.
Emily Blumberg, University of Pennsylvania. I think it's time to consider a stepwise reclassification of these tests, specifically screening tests that may be used to improve access to diagnosis. I do not think these tests should necessarily be ones that will be used to determine treatment, but rather to identify at-risk populations. And so would focus on that specifically to improve access, but ensure that adequate safeguards are in place so that people do not then see these as final results, but really an entry into healthcare.
Can I ask you a follow-up question to make sure that we get back to this?
Sure.
And do you feel like there is insufficient data to allow us to create special controls and mitigations for diagnostic tests?
You mean for I guess I'm concerned about providing adequate guidance for interpretation, specifically to guide treatment. So I'm a little less comfortable with that.
So you think there aren't data based on the Class 3 tests yet that these are
insufficient data to me.
Okay, that's fine. I just want to make sure I'm clarifying that. Dr. Prokop?
Yeah, I'm supportive of moving these to a Class 2. I think there is sufficient data. I'm going to echo Dr. Petty from earlier. We've been doing these tests for a long time. And so I think if we look at that data, the FDA will be able to put in sufficient safeguards for both types of tests, screening as well as molecular diagnostics.
Thank you, Dr. LaHose.
Hi, this is Ricardo Laos from UT Southwestern. I think the tests have a great track record with great performance, diagnostic performance. The safeguards can be placed. Comments can be made about how to interpret the tests. I think that a proportion of patients that may be infected with hepatitis B may not have access to confirmatory testing, and I think that can also delay initiation of therapy. So to me, if the diagnostic performance can be kept, I think the benefits, there's always, everything in medicine is a pro and a con. So initiating therapy in rural areas where patients may not be able to drive to somewhere where there's confirmatory test is something to consider. So why not keep the same diagnostic performance and proceed with the classification and use them as the class 3 are currently being used is an important consideration. I think the possibility of misclassification of the results will continue even if we keep them class 3, and that is a issue that also needs to be considered, while I think that reclassifying them can potentially improve the access to care and the net benefit be more significant. Those errors are going to continue despite the reclassification, unfortunately.
Thank you. Dr. Caliendo?
Angie Caliendo, Brown. I am comfortable down classifying all 4 categories of tests. I think we have adequate information and experience, and we've discussed appropriate special controls.
Thank you. Dr. Beavis?
Kathleen Beavis, University of Chicago. Ditto what Dr. Caliendo just said. And, as a pathologist, I've seen the evolution of these tests over the last, 25, even 30 years. And, they're so close. They've just gotten so much better that I'm, very confident that there is the information available to the FDA to be able to down classify these and put on the special controls.
Thank you. Ms. Schwartzett?
I do agree that we should be moving towards class 2 designations. As a patient, I'm thinking about my population. And I do not have hepatitis myself, but I do have liver disease. And I'm lucky that I'm okay, but others are not. What we really should be thinking about is the people that aren't getting diagnosed and that are going to develop all these higher level stages of liver disease because lack of diagnoses. And there are very few treatments for people with liver disease. Not everybody's going to qualify for a transplant. There are very few medications. And a lot of these people end up dying from this. That's what the real risk is. If they're not diagnosed, if they don't receive early treatment, this is what's going to happen. As for all these tests that are currently out, the risks are very low. I think labeling is key. I think it needs to be clear, not just for the medical community, but if somebody's taking home one of these tests from their pharmacy, they should have, specific and clear labeling. But the important thing is to get these out to the general public, to the people that are, can't get the care that I can get, that we can get on this panel. And so whatever is the easiest mechanism that's safe and effective should be available.
Thank you. Dr. Pereira?
Marcus Pereira, Columbia University. I agree with Dr. Caliendo and Dr. Lawas. I would support the reclassification of all of these devices to class 2, provided that those parameters in terms of test performance maintain a high level, such as what they are currently stated at. I don't think that, is what Dr. Lawas says. I agree that, the misinterpretation of these results is currently happening, and switching it to class 2 probably will not deteriorate that interpretation. In fact, actually, by instituting special controls, in particular when it comes to labeling and making sure that either the providers or even patients themselves are given the information necessary how to appropriately interpret and take the next steps with these tests, might actually enhance interpretation. Obviously, that would have to be carefully worded, but there is a potential here for actually improvement. Obviously, the underlying concept here is that improved access is a greater benefit than any of the risks for these tests.
Thank you. Mr. Spring? Oh, I lost Mr. Spring.
Sorry. Yeah, I clicked it twice. I also support the down classification. I think the going to class 2 will increase patient access, but probably just as importantly, will help the continuous improvement of those tests that are currently on the market today. And then one final comment, I know we're talking about US patient populations, but global regulators do follow and closely watch FDA's lead in these areas. And I think it'll encourage future down classifications in other countries around the world and increase access there as well.
Dr. Petty?
I also support the recommendation to reclassify class 2 for all the assays listed with special controls.
Thank you. Is there anyone we haven't heard from? Dr. Winston?
I concur. If it was not clear before, I just wanted to be very clear that I agree with the direction.
Dr. Cotton?
I also just want to give my support for downgrading. Thank you.
Dr. Walker?
Dr. Walker, I also concur with the discussion and the agreement to downgrade to classification 2.
Dr. Chu, did we hear from you?
Yeah, no, I agree with the, I concur with everyone else about downgrading to downgrading, down classifying to 2, but as long as there are these, as we've discussed, like the special controls are implemented and they're assay specific, especially my remaining concern is still with the quantitative assays. But as long as there are appropriate special controls in place, I'm in favor of downgrading all 4.
Dr. Moore, did we hear from you? And Dr. Moore gives us a visual thumbs up, which I'm going to put into the record verbally.
All I can say is I concur with the discussion. I have nothing to add.
Okay. Have I missed anyone?
My hand is up.
Oh, sorry, Dr. Ng.
Yes, I just want to say the answer is yes.
Okay. Anyone else? Okay, I think I can summarize this one for you, Dr. Stencil, pretty quickly. The panel in general believes that it would be appropriate, based on the amount of data that we currently have about how these tests perform, to consider starting the process to downgrade them to class 2. The panel is not concerned, but is adamant that the special controls do need to be in place that dictate the high level of sensitivity and specificity, and also that special controls will need to be in place to mitigate any potential risks of using these in certain patient populations, for example, those who have been recently vaccinated, and to distinguish perhaps between diagnostics and screening. And that for quantitative assays, there are special controls that cover limit of detection, limit of quantification, and linearity, so that we know exactly what these results mean. And I think that that's the overall consensus of the group. I didn't hear any that had strong feelings against starting the process of down classification. Did you have any other information that you were looking for from the panel?
I think we got what we need. I want to really thank the panel on this analyte, and we take seriously the charge to make sure that special controls in the down classification process are carefully worded and take into account patient safety. Thank you.
Excellent. There is one remaining question before lunch, so don't get your hopes up too quickly. So let's look at question 4 that I think people were concerned about along the way. So currently there's no authorized test for detection of and quantitation of surface antigen for HBV. So would you please discuss what you would think the appropriate intended use for such a device would be, discuss potential risk for both detection and quantitation of hepatitis B surface antigen, and whether or not you think there's enough data that we could build special controls and put measures in place that would mitigate risks to health. So if I could have input from the panel, I know that some people have already been talking about quantitation, and one of the questions that was already raised is whether that quantitation would be similar across all assays, so that we would have some consistency of the meaning and interpretation of those quantitative values. I think that was one of the ones that was raised. Dr. Ng?
Thank you. I think I would like to first state that quantification for me for a meaningful result would not be with surface antigen. It would be with a NAT test. I want to know a viral load because that's what I would monitor the effectiveness of treatment. I think the question I would want answered if we are going to work on quantification assays is what is the relationship of an antigen assay to a NAT? I would want to know if I pick it up on an antigen assay, would I always have a measurable viral load? And then I would want to know how to interpret it when there's a discrepancy. And finally, buried in all of that is how do these antigen assays perform with the various variants?
Great questions. Dr. Moore.
Yeah, Dr. Moore, University of Kansas. I have I apologize for this, a simple semantic question. Can I ask for clarification? Is this the detection, are we talking about two separate, or a single test that is detection and quantitation, or detection and then also quantitation?
Yes, the second.
Okay.
So potentially two separate assays or one assay at the same time, I mean, it could go either way, but don't think they have to be bundled.
I understand. Okay. Well, if with that said, I agree with the previous comment that the quantitation of hepatitis B surface antigen is not useful to me clinically. The PCR-based DNA quantitation is really the best, and really just the presence or absence of the hepatitis B surface antigen is all I'm really interested in.
Okay. So you would like a qualitative assay only? So a yes, no.
Yes.
Surface antigen. Okay. And one of the parts of this question, this was a complex question. I'll just remind you that one of the parts were, what would the intended use for such a device be? And since you've said that this might be of use to you, what would the intended use be, and what are the potential risks of an assay such as this?
Well, the use would be as a screening tool to clarify who has active infection, who doesn't, and then from there use that as a springboard to then launch to the hepatitis B PCR. So to me, it would be used as a panel as part of a serologic test, screening test, and there I think it would be beneficial to have just to extend the discussion further. Make it as accessible as possible within limitations, obviously. With regard to the qualitative aspects, the screening tool.
Mm-hmm. And as long as we're still on you, are there any specific risks to this test that you would like to mention that need to be mitigated?
Right. Well, the risk is just having a false positive,
Sure.
and then you'd have to have some caveat that states that confirmatory testing would be is recommended. Although, honestly, the performance characteristics of the test to date are pretty darn good, and the rare, false positives are quite rare.
Dr. Caliendo.
Angie Caliendo Brown. It's kind of interesting. Could this test be a inexpensive version of a NAT? I don't know the answer to that, and that's what I would like to know. And I think Dr. Ng was kind of bringing that up. How does this relate to NAT? Could you use it as if it's negative, it's useful because you've treated below? Could you use it as an inexpensive NAT? You'd need to know its LOD and how that compares to nucleic acid testing. I don't even know if there's an international standard for antigen, for Hep B antigen. So that's I'm not thinking about it so much as for screening. I'm thinking more of it as an inexpensive alternative. So there are risks, right? If it's in that situation, if it's not adequately sensitive, it's a problem. Because that's my only experience is this was hepatitis C antigen that never got sensitive enough. I think the correlate was 10,000 before we got a positive antigen. So clinically it wasn't useful. And so that's to me, that's kind of one way to think of the test. Could be useful if it actually worked, but I don't know that there's enough information to know if it could actually work as a substitute.
And I know Dr. Stencil needs to pop in, but I want to ask you a clarifying question before we leave you, Dr. Caliendo. Are you saying that you would see this as a tool for monitoring people on therapy?
Potentially. I don't have enough information to answer that, but I'm seeing it that way, potentially, yes.
Okay, Dr. Stencil.
Yes, thank you. Following Dr. Moore's comments, I just wanted to clarify the role of the FDA, at least in CDRH, the device center, and submission. So we only in narrow circumstances consider clinical utility. That usually is combination devices like a companion diagnostic. But for something like this, we look at, is it safe and effective either as screening or diagnostics? So there is nothing preventing somebody from developing a quantitative surface antigen test and submitting it to the FDA. And it's just a question whether it would be a class 3 submission or a class 2 submission. And as long as it's safe and effective and it, in either classification, then we could move forward to authorize it. But we largely stay out of the practice of medicine. And clinical utility, that's something that insurance companies and payers consider, but largely not the FDA for these kind of devices.
Okay, and Dr. Garcia, I'm trying to let the FDA go in response to previous before we move on to other panelists.
Sure, yeah, to answer Angie's question, there is a surface antigen international standard. And I know there's been a lot of questions in the field and also among manufacturers, like how is this test going to be used? And why is that question coming up? Because that's going to drive what the clinical study is going to look like and what the intended uses are going to be, which is why we're looking for your thoughts on the quantitative surface antigen tests.
Dr. LaHose.
Hi, this is Ricardo Laos from UT Southwestern. I think there are two potential uses, but the most important message I would like to send is that I think we need more information. But the first one is the hepatitis B surface antigen mutants and occult hepatitis B, where there may be a lower production of hepatitis B surface antigen. And how can we use the quantitative assay to better classify those two scenarios versus a surrogate marker of treatment response, as it's been alluded by Dr. Caliendo, but I am unaware of information to really help us guide in that regard. So I don't think we have all the information.
Thank you. Dr. Bloomberg?
At the risk, Emily Bloomberg, University of Pennsylvania, at the risk of repeating what other people have said, I would just say that based on the comments that have been made, many of which I've been thinking about as well, I really don't think we have sufficient information at this point to move this forward in the same way as the other tests. There are many circumstances where you could imagine it may ultimately be helpful, but the practical use of this now is insufficient to move it forward. I think more data is definitely needed.
Dr. Petty.
Kathy Petty, HealthSpring Global. To follow up Dr. Garcia's comment with respect to the international standard and to follow on Dr. LaHose's comment, I know we do have that international standard. We also know that there is tremendous variability in genotypes in many of using these international standards for surface antigen. So if we do, well, not if, I do encourage us to explore this type of assay because I think it may be a useful tool, again, a tool in our armamentarium for hepatitis B treatment and decisions for treatment. But I think we do not have enough information. I think it's very important for us to maybe support organizations that want to gather information in this area, and in particular, pay attention to testing variability with the genotypes, like we've seen with our other hepatitis viruses.
Thank you, Dr. Cotton.
So I was wondering if anyone on the call had expertise regarding whether or not NAT testing misses certain variants. And I know that sometimes surface antigen, there can be a gap where surface antigen is negative and nucleic acid testing is positive. But are there incidences where, due to mutations, nucleic acid testing doesn't pick up certain variants, such that we would really want a really useful surface antigen? Does anyone know of that situation?
Dr. Garcia?
Sure, yeah. So, I mean, we haven't seen any evidence to that effect that our tests are missing certain variants. That's not to say that doesn't exist.
Okay. I know it's an issue for some of the other nucleic acid tests that are out there, not for hepatitis B, but outside of hepatitis B. But I haven't really heard of it here. So as long as it's not a major issue, then that doesn't seem like we need surface antigen for sort of backup testing, the way we do use NAT testing for backup testing when surface antigen is negative, but we have a concern for active disease. Thank you.
Yeah, and to build on your point, there is, of course, the concern with the surface antigen and its detection of the different variants. And as Dr. Petty mentioned, the differences with the genotype detection. So that could definitely be an issue for the surface antigen tests that we would need to find a way to mitigate.
Dr. Chu.
Yeah, maybe I can just start by addressing kind of Dr. Cotton's comment. So, the hepatitis B genome has extremely low diversity, and the primers have been developed essentially. To my knowledge, I don't think there have really been, mutations that have been identified in among the sequence genomes worldwide for hepatitis B. And so, as a DNA virus, it has extremely low diversity. So, I'd be much less worried about potential escape from detection by nucleic acid tests versus, say, an RNA virus, such as HIV or hepatitis C. But that being said, I agree with the rest of the group. I feel that this particular test, the quantitative hepatitis B surface antigen, it's still looking for an indication. I mean, there have been some promising indications that might be useful for evaluating response to therapy in parallel with hepatitis B DNA, but I think right now we don't know whether this particular test has any advantage over simply running HBV DNA beyond the fact that it might be an easier test to run or a lower cost test. So I agree with the rest of the group that I feel that there's not enough information to downgrade or downclassify the from class 3 to class 2 for this particular test.
Dr. Ng.
Yeah, I just want to add to why I want to know the relationship, because you got that pesky problem of those empty virions, right? Like 100 to 1 ratio. And so how does that play out as an antigen test versus a NAT? I think I go with the NAT. That's my sense.
I'm going to throw in one more question for the panel before we try to wrap this up, because we're hearing a lot of the same thing, that there's not enough information and that we don't know how this would be utilized or where it would be adopted. And those are all fine questions, but I want to just kind of restructure the question is, do we have enough information to say what the special controls would need to be or what the labeling would need to be if such a product were developed? So whether or not it should be developed is kind of a different question. And it may be not. But if somebody wanted to develop it, do we have enough information to say that based on that decision tree, that we have enough information that we can actually develop special controls for a test like this, so it should be put into class 2 from the get-go, or do we not have enough information about what the risks are that we need to mitigate using those special controls, and so it needs to be started at class 3? I think that's a question I'd like to hear the panelists sort of address. Dr. LaHose.
This is Ricardo Laos from UT Southwestern. I don't think we have, or I'm unaware of enough information to say that we can have special controls to classify.
Thank you. Anyone else? I'm seeing many people nodding their heads. So I assume this is a consistent response from the panel. Does anybody want to add anything, or shall I try to summarize? Seeing no hands, I'll try to summarize. So Dr. Stenzel, with regard to question 4 about whether or not a surface antigen qualitative or quantitative test should be class 3 or class 2, the panel doesn't feel like there's sufficient information to help develop special controls. And so at this time, we don't have enough information to support moving this to class 2. So if someone does present data to the FDA about a test such as this, it should be evaluated under a class 3 mechanism according to the information that we have available to us at this time. Does the panel, can I see nods? I think that was a pretty united consensus. Does that give you the information that you need, Dr. Stenzel, or do you have additional questions?
Yes, it does. And we thank the panel, and we just wanted to be complete in our questions. And we understand that this is the panel's opinion as of today, with the information we have today. Thank you.
Okay, we are nearly perfectly on time. We have one presentation that was scheduled for before lunch from the FDA, but we're going to move that back and let everybody take a break because we've had some really good, strong, robust discussions. And so we'll take our lunch break now, and we will come back on the hour. So we will see you in an hour and 4 minutes. Thank you, everyone. Welcome back. It's now 1 o'clock Eastern Time, and I would like to resume the panel meeting. We're going to invite the FDA to start the next presentation. I would like to remind public observers at the meeting that while the meeting is open for public observation, public attendees may not participate except at the specific request of the panel chair. FDA, you may now begin your presentation.
Hello, my name is Ryan Karsner. I'm an infectious disease physician and the Deputy Assistant Director for Hepatitis and General Viral Infections branch within the Division of Microbiology Devices. This presentation will discuss the potential reclassification of qualitative serology-based parvovirus B19 antibody in vitro diagnostic devices from Class 3 to Class 2 with special controls. The purpose of this panel session is for the panel to discuss the potential future reclassification of parvovirus antibody assays. FDA is ultimately seeking recommendations from the panel members and the public on whether sufficient information exists such that the development of special controls, which along with general controls, could mitigate the risks from these devices such that the devices would provide a reasonable assurance of safety and effectiveness and therefore can be eligible for a Class 2 designation. The intended uses for these parvovirus antibody assays are for the detection of IgM antibodies and IgG antibodies as evidence of parvovirus B19 infection and may be used as an aid in the diagnosis of past or current infection with parvovirus B19. The tests are labeled such that a clinician should consider the results of these assays as presumptive for risk of fetal infection with parvovirus. The test may be used as an aid in the diagnosis of fifth disease or erythema infectiosum. Regarding the public health burden, most individuals are infected in childhood, resulting in 50 to 80% IgG seroprevalence reported in serosurveys. However, much of the public health burden largely rests with a few specific populations. Chronic or reactivated infection can be associated with increased morbidity for high-risk populations, such as immunocompromised patients or those with hemolytic anemia. The virus can also spread through blood, and therefore a pregnant person is at risk of passing the virus to the fetus, causing serious complications due to severe anemia, such as hydrops fetalis and miscarriage or intrauterine fetal death. Risks to health are associated with failure of the diagnostic device to perform as intended, or errors in the use of the diagnostic device, or other reasons for false results and subsequently improper patient management. A false non-reactive result may cause spreading of the virus to other individuals through contact and thus present a public health risk. Parvovirus B19 infection is generally self-limiting and benign for most healthy individuals, but again, may pose a more grave threat of chronic or reactivated infection with associated morbidity for high-risk populations such as immunocompromised patients or those with hemolytic anemia as discussed. The virus can also spread through blood. So again, therefore, a pregnant person with parvovirus infection, but a false negative result is at risk of passing the virus to the fetus without the knowledge of the patient or the healthcare provider. This can cause serious complications such as hydrops fetalis and miscarriage or intrauterine fetal death. Even though there may not be a change in indicated medication administered in most circumstances per se, FDA considers knowledge of the result and subsequent appropriate follow-up with ultrasounds, for example, to be a risk associated with these devices. A false reactive result may lead to the tested individual to be isolated and monitored for a short period of time, which could cause psychological stress specifically, particularly in pregnant people for the reasons that we discussed. If parvovirus antibody assays are reclassified as Class 2, special controls would be written to mitigate the risks to health discussed on the previous slides. The goal would be to maintain consistent high performance across devices with similar intended uses and for individual devices over the total product life cycle. There are some specific considerations for parvovirus antibody devices and their validation. Specifically, there are very few parvovirus antibody tests currently on the market, which may lead to difficulty formulating a composite comparator during the clinical validation of a new parvovirus test. Low prevalence of parvovirus IgM, even in the intended use population, can lead to difficulty in enrollment of IgM positive individuals in a clinical and analytical studies. Confidence intervals around performance point estimates can therefore be quite wide, and therefore there can be some uncertainty about the performance of these tests, even in otherwise well-designed and/or enriched studies. There are, of course, additional concerns related to cross-reactivity and interfering substances, along with other similar considerations for any other antibody test. Regarding the additional considerations for the potential reclassification itself, manufacturers would no longer be regulated under the Class 3 paradigm, but instead under the Class 2 paradigm, which has fewer regulatory requirements. Manufacturers will no longer have to submit a PMA application for these types of devices, but can instead submit a 510K to the agency for review prior to marketing their device. A 510K typically results in a shorter pre-market review timeline compared to a PMA application, which ultimately provides more timely access of these types of devices to patients. Considering the probable health benefits of the use of these devices and the nature and known incidence of the risks of the devices, FDA on its own initiative is contemplating reclassifying these post-amendments Class 3 devices into Class 2. FDA believes that when used as indicated, parvovirus antibody tests can provide significant benefits to clinicians and patients, including making a serological determination of past, recent, or current infection with parvovirus B19 as an aid in the diagnosis of fifth disease, or erythema infectiosum, and presumptive risk of fetal infection with parvovirus B19. FDA's reasons for reclassification are based on the scientific and medical information available regarding the nature, complexity, and risks associated with parvovirus antibody assays. The safety and effectiveness of this device type has become established since the initial approval of the first parvovirus antibody assays in 1997. We therefore have the following questions and requests of the panel. 1. Please comment on whether you believe FDA has identified a complete and accurate list of the risks to health presented by parvovirus antibody assays. Please comment on whether you disagree with any of these identified risks or whether you believe any other risks should be included in the overall risk assessment of parvovirus antibody assays. Please discuss potential mitigation measures, controls that FDA should consider that could mitigate each of the identified risks. And 3, based on the information presented and future discussion at this panel meeting, please discuss whether, based on the available information, the panel believes FDA should initiate the reclassification process for these devices from Class 3 to Class 2, subject to special controls. Thank you for your time.
Thank you. The FDA has received no requests to speak during this open public hearing portion of the meeting. Therefore, we'll go to the next item on the agenda. At this point, we will begin deliberations, but does anyone have a question for clarifying purposes of the FDA about the presentation we just watched? Seeing none, I'll move on. At this time, let us focus our discussion on the FDA questions, which we just saw. You have copies of these questions in the packet of materials that you received in advance. I would remind you to each of you when you begin speaking to identify yourself so that we can facilitate transcription. If you would show the first question, please. This is going to be the same structure as what we went through with HBV, where we're going to first focus on risks and whether the risks that were described are inclusive and exhaustive. So do we have everything that we would like to see discussed as a potential risk? Are there any risks that were described that you think are not appropriate for parvovirus antibody testing? With that, if you want to raise your hand, we'll take comments. I know it's after lunch, but still, we must all have some thoughts. Dr. Moore.
Yeah, I guess I'm sort of curious what the impetus or what the reason is for seeking a lower or having this come up for discussion? Is there a paucity of tests for parvovirus B19? I see two manufacturers on the accompanying sheet. Are they? I'm not quite sure what the rationale is for downregulating it.
I'll let Dr. Stencil respond to that, but I'll also remind you that part of the FDA's charge is to have all tests classified at the lowest classification appropriate. And so at this point, do we have enough data to move it from a class 3 to a class 2 with appropriate controls? But Dr. Stencil, in case I didn't cover that well.
Dr. Vanderpool, exactly. We underwent a review of all class 3 devices in microbiology. And we looked at those that we felt, should be considered for down classification. Typically, but not always, class 3 devices do limit the number of options that are available to laboratorians, clinicians, patients. And it is our charge to make sure that the tests are appropriately classified. We've previously underwent extensive reviews and were able to down classify many class 2 devices to class 1. So this is just an ongoing process to make sure that when appropriate, we look at the possibility of down classification. Some of my FDA colleagues may have something more specific to add about this particular analyte.
And I think the panel's charge is really just to say the FDA has gone through this data and made some, put together data about it, but they want to verify that we don't identify any risks that they missed and or mitigating opportunities that they missed. So with that, Dr. Pereira?
Yeah, Marcus Pereira here from Columbia University. I imagine we might have some similar discussions with parvovirus as related to also our previous discussion on hepatitis B. And I think some of it will relate around what exactly are the data surrounding the performance of these tests. I in my practice, I don't often use it. Being an adult transplant ID physician, sometimes we use it pre-transplant. But I do get a sense that there is a significant rate of false positive results, particularly when it comes to the IgG, and some false negative results with the IgM. But in order for me to at least have a better understanding, would anyone here on this panel provide some data on the performance of these serological tests?
Great question.
So I can probably shed a little bit of light, at least on the data that we have at the FDA. So, the performance of the tests currently on the market, at least in the clinical validation that we reviewed at the time, the tests did seem pretty specific, or at least the negative % agreement with, immunofluorescence or Western blot, or just a comparator ELISA that was already on the market, tended to be over 96%. For some of the tests, the sensitivity of the IgM, specifically for acute infection in certain populations, maybe was a little bit lower than that, around 91% for those people. But it also depends on what is the comparator and how are you establishing clinical truth. But generally, in the general population of the validation for the tests that are on the markets, the positive % agreement with the comparator was between 97 and 99%. And then, I can also speak to the fact that we don't, we checked our databases that we have to track errors that are reported to the FDA, and there were not a significant number of these tests having reported false results to the FDA, for what that's worth.
Okay, thank you. I mean, I think my particular concern would be when it's used for screening during pregnancy. Obviously, the stakes seem pretty high in that particular setting. And if you're telling me that the IgM has potential lower performance during that particular group, it does raise some concerns and whether we could potentially have some special controls in order for that sort of high stakes situation. But I think we might have to discuss this a little bit more.
Sorry, I'm muted. Dr. Bloomberg.
Thank you, Emily Blumberg, University of Pennsylvania. Following on that, I do wonder how much of an access issue there is with this particular testing. Like, do we know whether there are areas, you know, as opposed to hepatitis B, where I think we would have to say this is a global concern and enormous number of people affected, many of whom are in resource limited areas where testing really is, access to testing is a concern. Do we have any data to share in terms of that? I assume in terms of validation of the testing that regardless, there would still need to be some way to validate any test regardless of where it's at in terms of that.
Dr. Stenzel.
Yeah, I'm not sure. The FDA doesn't always have information about access to testing. More options is better if they're good options. Again, the consideration of moving from class 3 to class 2 is, you know, are there special controls that can mitigate risk? And as Dr. Paul raised, that we are looking whenever possible to down classify. This does intend to provide more options for tests that laboratorians and clinicians and patients can choose from, and it's our belief that that's a good thing as long as we can mitigate the risks. Thank you.
Dr. Prokop.
Yeah, I share some of the same concerns that Dr. Pereira raised, and I think it all depends on use case. You know, unlike you know, hepatitis and HIV, I don't. It hasn't been my experience that individuals with positive antibody tests automatically get a PCR. You know, it's kind of, does it fit clinically or doesn't it? So yeah, I really don't have a great concern about, you know, some misses of fifth disease, but like you said, during pregnancy, aplastic anemia of immunocompromised hosts, I think that's where the special controls might be particularly applicable to how you're using the test. And in those situations, you may want to follow up with a more definitive and nucleic acid test in a positive that could be a false positive. I'll stop there.
Thank you. Dr. Lowe's?
Yeah, I think I think multiple considerations. My understanding is that in pregnancy, what we are going to be focusing on is the risk for hydrops fetalis, which if the pregnant female already has a positive IgG, means that there's no risk. So at least from the numbers that we've been quoted for the performance of the IgG, it appears that it may perform well in that scenario. I think, again, pregnant females are going to be in different areas of the country where potentially more options for testing could be helpful. On the other hand, in the immunocompromised host, we do see pure red cell aplasia from parvovirus B19, and the IgM is a unreliable test to diagnose the infection in immunocompromised host, and it often requires a PCR to complement the diagnosis, as well as, you know, chronic arthritis in that population may also benefit from a PCR. And already the IgM in at least one of the tests that's quoted here in the materials that we have may have a PPA of 78%. So, and maybe my only question for the FDA is, what is the track record overall of these assays? You know, similar comments than what we made to hepatitis B. I mean, what's historically been these tests? It appears that one was approved in 1999 and the other one in 2017. So there may be some information.
Dr. Petty?
Kathy Petty, HealthSpring Global. Following on what Dr. Prokop and Dr. Lajos had said, special controls, I think, are very important if we reclassify to class 2. I do use parvovirus serology testing in the non-immunocompromised hosts, and often do the acute and convalescent pair sera. So perhaps in product labeling, giving some guidance that, again, used in the aid of the diagnosis of, could be very valuable, particularly for those institutions where it could take a week to 10 days to get a parvovirus viral load back to clinicians who don't have access to viral loads in a rapid fashion.
Thank you. Dr. Caliendo?
Angie Caliendo Brown. So I think the FDA has outlined appropriate risks. I don't have anything to add to that, but I do find it very interesting that almost everybody has mentioned PCR for parvovirus, and there's not even a test available. So that's through the FDA. So maybe one of the mitigating measures needs to be the FDA finding a way to maybe use a de novo 510K to help companies get a quantitative parvo PCR out there, because that's the test many of people are using clinically. So that's that. I don't have anything to add to the risks. I think they're covered, but when we get to mitigation methods or mitigation suggestions, that would help, I think, put a clinical context on a result.
Thank you. Dr. Karsner?
Yes, I just wanted to address the question of the track record for these tests. So the information that we have is the performance and the validation that was included in your packets, as well as the kind of post-market evaluation, or at least reporting systems that the FDA has, where we have not really received reports of false results for these assays. So that's the information that we have about the track record for these tests.
Dr. Stencil?
Yes, and I wanted to respond in particular to Dr. Caliendo's comments that there is no approved PCR test. Part of what we're doing here is looking at, is this ready to be, down classified in general, and that we hope that invites, if it can be mitigated and it can be safe as a class 2 device, we believe it would encourage developers to take a look at this. I do have to state that even if we down classify and we have all the measures in place to make sure it's safe and they're appropriate, the FDA does not develop tests, and we can't compel people to come in with their submissions. What we can do is make sure that the classifications are as low and appropriate as possible, so that it's as easy as possible, should we say, for developers to come in. Thank you.
Angie, did you want to say
Angie
something?
Caliendo Brown. So Tim, thank you, because I think that's where the real value here is, right? Is if we can down classify, and that gives you the ability to make it easier for a PCR assay to come through. You're right, we all know we can't beg, borrow, and steal sometimes to get manufacturers to make tests, but that is probably the real value here. So thank you for that clarification.
And I have a clarifying question for Dr. Stencil as well, and that is, this topic was focused on parvovirus antibody tests. Is it a potential for the panel, based on some of our discussions, when we get to question 3 about, do we recommend down classification? Could we recommend that down classification is more broad than just antibody tests, but any parvovirus tests? Because I think that's one of the things maybe I'm hearing as I'm making notes.
Tim Stenzel, yes, that would be very helpful. It would be difficult for us to, receive a DNA-based test and say it's okay to be class 2 while we have these other tests that are class 3. So that would be confusing to developers in the community. So that would be extremely helpful. Thank you.
Excellent. I think that gives us some guidance. I'm going to just go back and see if I can wrap up question 1, which was about risks. I heard a couple of people say that they think all of the risks have been included and are appropriate. I want to give everybody one last chance in case there are any risks that they would like to speak of that we haven't mentioned that aren't included in the FDA's considerations. Dr. Chu.
I'm sorry, I was on mute. Sorry about that. Yeah, I just wanted to comment that I feel that the FDA has adequately described the risks associated with this particular test, but the only comment that I had was that I feel that the special controls have to emphasize kind of the intended use, because I would say like an identified risk would be that the test is ordered inappropriately. In addition to that, the test is the test interpreted or the results are maybe incorrect. There would be a risk of kind of inappropriate use of the test, and so providing specifics regarding the intended use population and regarding the clinical use cases, as others have alluded to, I think would be really important.
Any other comments about risk? Okay, seeing none, I'm going to summarize. I think that the panel in general feels like the risks described by the FDA are appropriate and that they've captured all of those risks. The concerns that were raised by the panel were whether or not those risks would perhaps also needed to include special controls for special populations and getting back to intended use. So I think this is true, especially for use in pregnant women. I think that also probably there need to be risks that maybe are associated with ordering appropriately, especially as it relates to IgG versus IgM, because those have very different interpretive values. And potentially that the risks of false negative, false positives apply as well to other parvovirus types of testing, which would include molecular tests for DNA. So, Dr. Stencil, does that give you the information that you need for question 1?
It does, and no follow-up questions. Thank you.
Perfect. Okay, so then let's move on to question 2, which we've already talked about a little bit, which is what are the mitigations and special controls that the FDA should consider in order to address those identified risks. We've already talked about making sure that there's appropriate labeling to clarify intended use, clarify when it's appropriate to order the different tests in different populations, and how to interpret the tests. Are there other mitigations that we need to consider as well for parvovirus testing? And remember that we can be a little bit more broad and include mitigations or special controls that we think would be appropriate, excuse me, for DNA-based PCR type testing as well as for antibody testing. So if anybody has any other special controls other than labeling and intended use statements and making sure that sensitivity and specificity are set at appropriate levels. Dr. LaHose?
Hi, this is Ricardo Laos from UT Southwestern. I don't know if this applies to the question, but transplant recipients often receive IVIG as treatment for rejection or donor-specific antibodies, and it is always challenging to interpret serological assays in patients that have received this type of products. And then, as I mentioned before, the performance of a serological assay in a immunocompromised host. But I'm unsure that that really answers the question that you were with that copy.
I think it does, because I think you're saying that here's a population that needs special intended use comments or labeling comments about how to interpret those results. And this may be the point at which you say that one of those mitigations is reflexing to a molecular test, right? As opposed to just making a treatment decision based on an antibody test alone. Other mitigations, Dr. Ng?
Well, of course, you open that door, Bobby. In situations of immune perturbations, and that includes pregnancy, what is known about reactivation of latent disease? And then if we're going to bring in viral loads, What's the relationship of a viral load to infection and transmission versus just reactivation without clinical consequence?
So the relationship of viral load to clinical outcomes?
Yes, what's the clinical usefulness of a viral load? Is there a threshold that tracks with infection and transmission?
I'm going to pass this off to Dr. Caliendo, who's more knowledgeable than I am.
No, I can't answer that question, and I don't know that anybody can. And I think that's one of the questions that needs to be addressed, which is why a molecular assay to be clinically useful needs to be quantitative.
Yeah, I'm not sure that it does.
Because there is this low-level reactivation. I haven't seen data, Valerie, on exactly what that threshold is, and it's probably different in different patient populations, but that's a clinical question that needs to be addressed, and that would be, I think, the test as opposed to a qualitative assay that might help. I mean, it depends if you're testing amniotic fluid versus blood, right, and what the patient population is. But I think it's very interesting, which makes my special controls is I think molecular is you would have the same performance characteristics that you would of any viral load assay. And it may be one of those that we need to test so that we can actually address some of these important clinical questions. It is interesting that you could put as a special control to reflex to a molecular test when there isn't one available that's gone through the FDA process. So is that okay to do? I guess my question.
I mean, I think that we're just throwing out ideas and possibilities. I don't think that the agency would necessarily, just because we've said that in this panel discussion doesn't mean that will be one of the special controls.
How is that even possible that we would say something and they wouldn't absolutely do it?
I don't know. For years, I've not known that. Yeah. Dr. Karsner, would you like to provide clarity where I have provided muddiness?
Sure. Well, I'll just address two things. One is that, generally for a lot of our serology tests that we regulate, the labeling includes limitations about, caution in interpreting serology test results in immunocompromised individuals for the reasons that we've discussed. And just to sum it up, I think it has to do with the etiology of immunocompromise can be very different depending on who the person is, what's going on, what medications they're taking, et cetera. And then the degree of immunocompromise. We don't always know how to interpret serology results in general in those populations. And then the other question about trying to interpret a viral load in respiratory diseases is quite challenging. I think, we borrow the idea of viral load from diseases like HIV and CMV, and HBV, for example, where we have kind of established for some of them, medical decision points at certain concentrations in blood. But I think we saw with COVID trying to interpret a pathogen burden in a respiratory specimen as it relates to infectiousness is really challenging, also because infectiousness has to do with the amount of time someone is exposed and how good is the ventilation in a room and all those sorts of things. So I think there are a lot of challenges with that.
Dr. Caliendo.
Angie Caliendo Brown. Let me just clarify. I wasn't talking about the respiratory compartment. I was talking about blood.
Oh, okay.
So, so just because I agree totally with what you say with respiratory, but just to clarify.
Okay.
And I think the larger concern is about vertical transmission, right? I mean, that's what we're kind of focusing on thinking.
I see. Okay.
Any other mitigations other than labeling and, indications about appropriate use? Again, keeping sensitivity and specificity at higher levels, which I think the FDA has data for already. Okay. Seeing no more conversation on this, I will summarize that I think in general, the panel agrees that special controls, we understand probably what those need to be, and they need to be population specific, and they need to include labeling, and they need to include appropriate sensitivity and specificity performance measures, especially understanding specificity of IgM as well as sensitivity of IgM, because that's going to perhaps be used more often during pregnancy. But all immunocompromised patients need to have appropriate warning labels and limitations to interpreting these results. I think that that pretty much covers it. And I think that the panel didn't have specific concerns about what those labels or limitations should look like. Dr. Stencil, did you need more information from the panel?
I think that's very helpful, and thank the panel. And no follow-up questions. Sometimes biology limits sometimes what you can do. And that doesn't mean you don't test. It just means you need to understand the limits of that test and make the appropriate laboratory and clinical decisions. Thank you.
Okay, so this moves us on to question 3, which is always the fun question. So if we could get the panel to summarize basically if they believe there's enough information to allow reclassification of these devices from class 3 to class 2, subject to special controls and other mitigating processes. So with that, if we can kind of go around the room, if you'd like, I'll just call on people, or if you'd like to raise your hand. There we go, Kathy Petty.
Kathy Petty, HealthSpring Global. I think we should again just go back to refocusing our conversation on the technology and the experiences and data that we've had thus far with serology testing. We all have used it a lot in clinical practice. We all understand that a single point in time test may not be indicative of a diagnosis, and that repeat testing, either acute or convalescent or reflex to a molecular test, is commonly used in practice. So I think with the data available to us now, I believe reclassification to class 2 is safe and would be effective.
Dr. Caliendo.
I agree. Oh, Angie Caliendo Brown, I agree.
Okay, Dr. LaHose.
Ricardo Laos from UT Southwestern, I agree.
Dr. Beavis.
I agree as well. Kathleen Beavis, University of Chicago.
Dr. Pereira.
Marcus Pereira, Columbia University. I agree as well. I actually think reclassifying as class 2 would be a great progress. It sounds like we don't have enough tests for parvovirus, and actually lowering the threshold would potentially lead to more tests becoming available.
Mr. Spring.
Yes, thank you, Brad Spring with Roche Diagnostics, and I agree with Dr. Pereira's comments there too. I agree with down classification. I think it would open up the market more for more of these tests to come into play.
Dr. Chu.
Yes, Charles Chu, UC San Francisco. I agree with down classification.
Thank you. Dr. Blumberg.
Emily Blumberg, University of Pennsylvania. I agree with down classification.
Dr. Wensenson.
Nicholas Vincenzen from NCI. I agree with down classification.
Dr. Walker.
Dr. Roblina Walker, I also agree.
Dr. Moore.
I think Dr. Petty summarized it beautifully, and I have nothing to add. I agree.
Dr. Cotton.
I also agree with downgrading.
Dr. Ng.
Can't we just do a thumbs up?
Sure. Everybody want to do a thumbs up instead of
That'll be a lot faster, right? This is not controversial. Thank you.
I don't know why you didn't tell me this 5 minutes ago. All right. You want to thumbs up me?
Well, does that work with the recording?
Oh, well, since we're not voting, I think it's fine, because I can say that we have a true consensus here. We don't have any concerns to share with the agency. Everybody is generally supportive of the fact that there are sufficient data to help develop special controls and labeling, etc. So we would strongly recommend that the agency consider down classifying for parvovirus antibody tests. Dr. Stencil, do you need any more information from the panel about this topic?
Well, I just want to make sure that if somebody wanted to say something and not just put thumbs up, that they're welcome to do that. This is the time for the FDA to get input, and we really do value that input. And so, with that, we have what we need. But if someone wants to say anything else, I see there's a few hands still up. So we welcome that input as well. But I defer to you, Dr. Paul.
Let's see, Dr. Cotton.
Oh, sorry, my hand was up. It was astray, sorry.
All right,
I usually leave myself on mute and leave my hand up and my thumb up, so I'm right there with you. Okay. Does anybody want to add anything to this conversation? I think this one was a fairly easy one for us to think about, so that's probably why the conversation was a little bit less intense. So, okay, at this time then, we are ready to begin our third and final session of the day. I'd like to invite the FDA to start their final presentation. Again, I'm going to remind public observers at this meeting that while the meeting is open for public observation, public attendees may not participate except at the specific request of the panel chair. FDA, you may now begin your presentation.
My name is Noel Gerald. I'm with the Division of Microbiology Devices in OHT7 OPEC. And I'm going to talk to you about potential M. tuberculosis interferon gamma release assays device reclassification. So the purpose of this meeting is to discuss the potential future reclassification of Mycobacterium tuberculosis, which I'll refer to as TB, cell-mediated immune reactivity in vitro diagnostic devices, such as interferon gamma release assays, which I'll refer to as IGRAs. FDA is seeking recommendations from the panel members and the public on whether sufficient information exists such that the development of special controls, which, along with general controls, could mitigate the risks from these devices, such that the devices would provide a reasonable assurance of safety and effectiveness, and therefore can be eligible for a Class 2 designation. I'm going to talk a little bit about the public health burden of TB. You can find these details and a lot of additional details about this in the executive summary. TB case counts and incident rates have steadily decreased in the US over the past 30 years. 8,300 reported TB cases were in 2022, which is 2.5 cases per 100,000 persons. Case counts are returning to pre-pandemic levels following declines in reporting during the previous 2 years. CDC estimates that up to 13 million people in the US could have latent TB. The worldwide TB burden is estimated at 10.6 million persons, and globally 1.6 million people died from TB in 2021. As a few additional details, pulmonary tuberculosis is the most common clinical presentation of tuberculosis in adults. And infection occurs by transmission of the organism through inhaling airborne particles that contain MTB that are released from individuals with active pulmonary disease. So most people who are infected with TB are asymptomatic, which is known as latent TB infection. And the latent infections are not contagious and do not result in clinical disease in most cases. But overall, there's a 5 to 10% lifetime risk for patients with latent infection to develop active TB disease. And this risk varies to many factors, including immunosuppression. Of the United States TB cases, more than 80% are attributed to reactivation of untreated latent TB. There are numerous antibiotic regimens available to treat TB. However, adverse drug reactions are common, and patients should be closely monitored while on therapy. A little bit more about interferon gamma release assays, IGRAs. IGRAs are indirect tests for TB. They're in vitro blood tests which measure T cell release of interferon gamma following stimulation with TB antigens to aid in the diagnosis of TB. The commonly used ESAT6 and CFP10 IGRA peptide antigens are specific to organisms in the Mycobacterium tuberculosis complex, but they're absent from the BCG strains that are used in vaccines and most non-tuberculous mycobacteria. So IGRAs are useful in BCG vaccinated persons and in clinical scenarios where a single patient visit is advantageous. In contrast, the tuberculin skin test, TST, is another type of indirect test for TB, which requires a second patient visit 48 to 72 hours after administration. Additionally, it is known that prior BCG vaccination or infection with non-tuberculous mycobacteria can cause a reaction in the TST. So this is a slide with a mock intended use statement of a TB IGRA. And I'll just walk through it briefly here. We mentioned the assay name is an in vitro diagnostic test using a cocktail of TB antigens, which would be described to stimulate cells in whole blood, typically. The detection of interferon gamma by, and we would describe the specific technology used, is used to identify in vitro responses to these antigens that are associated with Mycobacterium tuberculosis infection. The assay is an indirect test for M. tuberculosis infection and is intended for use in conjunction with risk assessment, radiography, and other medical and diagnostic evaluations. So here are a list of the PMA-approved TB IGRAs. These are all qualitative tests. The first one was approved 22 years ago in 2001, and the technologies have continued to evolve for things like sample processing and test interpretation. This slide also includes some of the performance estimates that can be found in the approved labeling for each of these tests. As there is no true reference standard for TB infection that's applicable to all cases, sensitivity here is estimated as compared to culture in a population with confirmed pulmonary TB disease. And then specificity is estimated in a population of subjects with low risk of TB infection. There are other populations that are included among the clinical evaluations to validate these tests, and I'll talk about some of those a little bit later in this talk. These are the risks to health of inaccurate results that we've identified. False negative results, incorrectly operating a device causing false negative results, and incorrectly interpreting results as negative results can lead to progression of active or reactivation of latent TB disease in individuals, spread of disease in the community, or missed opportunities for diagnosing an underlying condition or disease such as HIV infection that may have been unrecognized and may be contributing to the progression to active TB. False positive results, incorrectly operating the device causing false positive results, and incorrectly interpreting results as positive results can lead to unnecessary treatment with then the associated drug toxicities for these therapies. Unnecessary patient isolation and radiologic imaging and laboratory testing, and unneeded contact tracing, resulting in wasted healthcare resources. So aspects of IGRA device validation, labeling, and use that should be taken into consideration when thinking of potential mitigations are discussed here. So labeling includes clinical performance in several different populations, which include, as I mentioned, patients at low risk for previous TB infection, in the absence of risk factors, patients with culture-confirmed or NAT-confirmed active pulmonary TB infection, patients at high risk of latent TB, and patients with a history of non-tuberculous mycobacterial infection or colonization, which could be cross-reactive. So the labeling includes the performance estimates. As I mentioned, these are, truly estimates in the absence of a reference standard. So specificity is estimated in comparison to the expected negative results for a population at low risk of TB infection. And sensitivity is estimated, from the population with active pulmonary TB. Labeling can also include, performance as additional information agreement to other indirect tests. So positive % agreement or negative % agreement to the results of the TST test or another IGRA. The labeling does, highlight, the limitations of studies conducted in the absence of a true reference standard. And it includes other limitations that are relevant, such as a negative result does not exclude the possibility of infection with M. tuberculosis, and noting that these tests should not be used alone, that they must be used in conjunction with each individual's epidemiological history, current medical status, and the results of other diagnostic evaluations. Some other things to take into consideration include the years of experience that clinicians have had at this point on, the recognizing the limitations of these tests and considering their appropriate uses. And then also the existence of guidelines from organizations such as the American Thoracic Society, CDC, and IGRA that explicitly discuss the appropriate use and interpretation of these tests. So lastly, some additional considerations for reclassification. Reclassification has the potential to increase opportunities for innovative diagnostic devices in this space. There would be a reduced regulatory requirement for sponsors and manufacturers. So PMA-specific requirements would be removed, but the belief is that the risks can be mitigated by special controls. So these are the questions for the panel. 1, please comment on whether you believe FDA has identified a complete and accurate list of the risks to health presented by M. tuberculosis assays, in this case specifically Interferon Gamma Release Assays. Please comment on whether you disagree with inclusion of any of these risks or whether you believe any other risks should be included in the overall risk assessment of M. tuberculosis assays. Please discuss potential mitigation measures or controls that FDA should consider that could mitigate each of the identified risks. Based upon the information presented and future discussion at panel meeting, please discuss whether, based on the available information, the panel believes FDA should initiate the reclassification process for this device from Class 3 to Class 2, subject to special controls. Thank you.
Are you on mute, Bobby?
It is always the best way to deal with me. We are scheduled for a 15-minute break now, and I think we'll go ahead and take that in case people need to stand up and refresh, and then we'll have our discussion about these questions when we come back. So if you could be back on camera in 15 minutes, that would be wonderful. Thank you. Thank you. I'd like to resume the panel meeting. At this point in the agenda, we have space for open public hearing, but the FDA received no requests to speak during this portion of the meeting, so we'll continue with the agenda. Before we begin deliberations about the TB IGRA assay, does anybody have brief clarifying questions for the FDA? Mr. Spring.
Yeah, Brad Spring with Roche Diagnostics, and not sure if everybody knows, you've previously downclassified other tuberculosis assays, correct? To class 2.
Yeah, this is Noel Gerald, Assistant Director of Bacterial, Respiratory, and Medical Countermeasures. Yes, we did reclassify molecular tests, so NAT assays, from class 3 to class 2.
Thank you.
Dr. Ng?
And so I just would like clarity. As I understand, these IGRAs are primarily used to identify latent TB, but the positive predictive agreement is based on active TB, correct?
So yeah, so they're actually, the intended use doesn't explicitly state latent TB. We understand that that's how that's a big part of how they're used clinically, and it's just part of that. Also, is it's very difficult to conclusively show that someone has latent TB until it's progressed to active TB. So it's part of the issue of when we say we can't adjudicate every single case of TB, and why we don't have a true reference standard. And so, it's why we've had to take this approach of, having these, kind of well-defined benchmarks that we can, kind of assess performance, and then, evaluating the performance in other important populations.
Dr. Chu?
Yeah, I had one question for the FDA. So one thing that was mentioned in the presentation is that, potentially by down-classifying this category of assay, it might potentially open the way for other kind of tests in that category. My question is specifically, are you referring to a category of, it's a very broad category of, potentially human host response biomarkers, or are you referring to, because that's the, that is what makes this test, I think, a little different than other tests that look directly for either nucleic acid or protein from the pathogen, but rather focus more on the host response. And so I'm wondering, is that what you're referring to, that it might open up assays in the future based on host response for infectious diseases?
Yeah, I'll take that. Noel Gerald, FDA. No, I don't think that we were specifically, indicating or meaning to imply biomarkers in general, right? So it is really specifically on, cell-mediated immune response assays. The same way we have humoral, like antibody, response. So that was the intent.
Dr. Cotton?
Camille Cotton, Mass General. I would just like to emphasize that this field will probably be growing. So we've had CMI tests for tuberculosis for a long time now, and I think it's definitely time to consider downgrading them. There are some non-specific immunologic markers that are cell-mediated immunity based, and then some specific CMV type tests. So I think the field is growing, and I think we should be sort of thoughtful about that, or at least I would encourage the FDA to be thoughtful about that, as that field is likely to grow. My second comment relates to the fact that these are lymphocyte-based assays, and I think many of us on this call who deal with immunocompromised patients experience a lot of issues with a lymphocyte-based assay, especially when they are used in patients on very potent immunosuppression and whatnot. It doesn't mean that they shouldn't be downgraded per se, but I do think that clinical context is important. We often see them being used to do things like rule out tuberculosis and other things that infectious disease specialists kind of come upon, where we need to do some educating. So it's sort of a new field. There are a bunch of caveats. That being said, I think it would be reasonable to consider downgrading, but it's a new field.
Okay.
I see my colleagues.
We're still in the phase where the only thing I'm asking from you right this second is if you have clarifying questions for the FDA. So if that is a true statement, then Dr. Hose?
Yeah, I think I just had some big picture comments and then answers.
If you have comments, let's hold those till we get into the comment
sorry.
section, but I'm just trying to make sure that everybody has a chance to ask the FDA what they are asking of us. So if you have any clarifying questions, feel free. Dr. Pereira?
Yeah, I think the data was provided in terms of, positive predictive and negative predictive. Were there any data on, in particular for the quantiferon test, in terms of indeterminate results, how often those come back? And I think that sort of it alludes to all the transplant ID providers over here that we often see that result. And we'll be talking about that in terms of sort of identifying some risks that perhaps special controls could be developed around. But I any data around sort of how often those results come back, or?
Dr. Gerald?
Yeah, so I think that for FDA, really the main information that we have is in the labeling, so the rate of indeterminate results that are observed there. I think what you're getting more to the question is, the follow-up, right? So these labels, are usually pretty explicit of what should be your next step or what you should be thinking of next when you get an indeterminate. So, I don't think that we have a good idea in practice of then what happens to that indeterminate, the next time someone does a test. So I think that's probably what you're asking, but I think mainly what we know about the rate of indeterminates is that first time and what's seen in those validation studies.
And I would point out to people that while in the data that the FDA presented, one of these assays has been was approved more than 20 years ago. It's actually the same assay with a different kind of parent company now that got approval in 2019. It's still the quantiferon assay. And so, I think that it's worth noting that we really basically only have 2 assays on the market for this purpose. And whether or not that makes any difference, I don't know, but just something for you to think about. So with that, let's focus our attention on the questions that they've asked of us. You have copies of the questions in your packages. I would ask that each of you identify yourself each time that you speak. So with that, question number 1, which I think we all know by now, is do you think that the risks are fully described? And do you think that there are any risks that are described that don't belong, that need to be removed? And the risks were false negatives and false positives, either from operating the device incorrectly, or just incorrectly interpreting the device, or just the device not performing as well as it might have. So do you think those risks are sufficient? Dr. Blumberg?
Sorry, I was having trouble unmuting. Emily Blumberg, University of Pennsylvania. I think the risks as described are accurate, but the issue, and I think Ricardo was talking about this and Camille a little bit too, is that there's so much that's population-based risk. And so quantifying the risk really varies based on the population you're doing this in. So clearly the risk of an indeterminate or inaccurate response is greater in immunosuppressed populations. It's also greater in people with active infection. And so I think that it's just important that we add to what was stated, that these risks may be greater in some of the populations where you're most interested in the answers, the immunosuppressed, the person who comes in with symptoms, because the tests as currently used are often inappropriately used to make a diagnosis of active TB.
Thank you. Dr. Hose?
Yeah, I think Emily made some of my comments. So I would say that for a disease that probably involves 1 out of 4 people living in this world, that's a latent TB, and probably the number 1 infectious leading cause of death, and now number 2 after COVID. It's surprising that we only have a few tests in the market to try and mitigate a disease that if we identify it on the latent state, we have pretty good preventive measures, meaning latent tuberculosis therapy. So I do think that the risks have been identified correctly. I'm probably going to repeat what's been mentioned before, but number 1 is that person that may receive a biologic or may just be being screened for latent tuberculosis for XYZ reason, but has a low pretest probability. So that's a population in which we need to be careful about false positives. Number 2 is the well-known fact that not everybody that has active TB is going to have a positive TB IGRA test result. That is a known fact. And then what's been alluded before is all those already immunocompromised hosts that have this test and may have a false negative test. But I do think that the risks have been adequately identified of false positives, false negatives. And I do think that there's ways to probably mitigate this risk by labeling, establishing standards in different populations, like it's been alluded to in the FDA presentation. And I think we need to try and make tests more available, because this is a disease that's quite preventable by correctly identifying those with latent TB.
Dr. Stenzel?
Yes, thanks for all the comments so far. I did want to state that the limitations in the labeling of these tests does specifically mention, care in the immunocompromised population. I also wanted to state something that may not be obvious to everyone. So, we have 2 providers in the United States of this important testing category. And even though they're approved, it doesn't mean their work is done. So there are more annual work to do to maintain these approvals. And then if they want to make any changes, it's more difficult to make changes for the benefit of patients and clinicians and laboratorians with a PMA. And then also, if they need to move or expand their manufacturing footprint, they need to submit that to the FDA, and then they need to be inspected. And having personally been responsible for 3 PMA moves in my career prior to joining the FDA, I can testify how difficult that is. So even though there may be a few, we also are concerned about the right classification to help them maintain their market presence and help ensure that they're continuing to be able to be available for use. Thank you.
Yeah, you would hate to see somebody drop their manufacturing because they couldn't deal with the regulatory environment. So yeah, that's a true statement. Did other people have comments about the risks as described? Were there any? We have discussing specific populations, particularly immunocompromised people and populations with low positive predictive values. Dr. Beavis?
Thank you, Kathleen Beavis, University of Chicago. Yeah, especially having heard what Dr. Stenzel just said, there's a real risk if these leave the market. In our ancient memory, when people used to get skin tests, even in a captive hospital population, when I was at Cook County and we were very aware of TB testing, over a third of employees wouldn't return to get their skin test read. And that is, a real risk if these tests do disappear from the market and, can't continue to be improved.
Dr. LaHose?
Yeah, I think Dr. Beavis makes a spectacular point because the difference between the PPD and these assays is that the PPD is very user dependent on who reads the assay. And I think there is a much higher risk of false positives because the area of induration with PPDs is misinterpreted by the area of erythema. So losing such a test when 1 out of 4 people living in this planet could have latent TB, could be a high risk.
Yeah, and right now we're focusing on risks of the test that the test causes to patients. So does anybody have any additional risks that they'd like to add, or any risks that were described that they think shouldn't be on the list? Dr. Pereira?
Yeah, Marcus Pereira, Columbia University. So in addition to the indeterminate results and sort of all of its misinterpretations, which should be included here as a risk, the only other thing I can think of is sort of follow-up testing, sort of repeat quantiferon tests. So if you've had a positive test and you were treated, sort of the potential lack of necessity in repeating that test. And that certainly, that could lead to potential unnecessary retreatments. So that could be a potential risk here if it's sort of not used in accordance in terms of what the test represents.
Okay. Dr. Petty.
Kathy Petty, Health Spring Global. One more risk is for those that are false positives and then a subsequent workup is required. We missed adding that then they are being delayed for treatment for their Crohn's disease or rheumatoid arthritis. So they're being delayed to treatment that could be associated with significant morbidity.
Okay. Dr. Cotton.
2 things. One, if we were to lose this testing and revert back to PPD testing, we would miss a huge number of cases of latent disease, especially in people on dialysis or with cirrhosis or any kind of major comorbidity. The PPD testing is a lot less sensitive, so that would really be, in my practice, pretty disastrous. I actually stopped doing skin testing about a decade ago because, yeah, as you nicely, as was nicely outlined, I think by Dr. Beavis actually, that people actually just don't come back for follow-up testing, which is a big issue. The second thing I wanted to highlight, and I'm not sure if it really belongs here, in a way it sort of doesn't necessarily relate to downgrading, but I just want to highlight, and I'm not sure how the agency is aware of this, but so with quantiferon testing, you get either positive, negative, or indeterminate results. And with T-spot, it's either positive, negative, or borderline. And that indeterminate tag is really problematic. When I think all of us on this call probably understand what it means, but when I give lectures on this subject, many people come up to me and say, don't you know that indeterminate means that they should get latent TB prophylaxis? And I give 9 months of INH. And so there's a huge amount of confusion. I'm like, oh no, it's like, it just means the test didn't work. And they're like, no, indeterminate means, I'm like, no, it doesn't. It just means, I said, it's the same as though they dropped the test tube. So in that it's not valid data, right? And so, yeah. So I just wanted to highlight that as an issue. It, in a way, it doesn't relate to downgrading, but for me, that's one of the bigger issues with this testing, especially because a lot of testing gets sent like 1 day after liver transplant. So it's always indeterminate. And then people like call me about that. So it would be helpful to have some guidance there, but that might be for a different day.
Dr. Gerald?
Yeah, just that I definitely acknowledge what Dr. Cotton just said. It is something that we've run into also when talking about equivocal for other tests, this kind of vagueness of use of the terms indeterminate, equivocal, or borderline. And it is something that we have been trying to be a lot more explicit about when we're talking with the sponsors going forward. So your point is very well taken.
Any other comments about the risks of the actual incorrect results? I think then that I will summarize. What we're hearing in general is that the risks were captured. We didn't have any risks that you showed that we thought should be removed from the list. However, the risks list could benefit by calling out specific populations in which those risk levels were higher as opposed to others in which maybe it was lower. Obviously, immunocompromised people, but also those people with active disease for whom the risk of a false positive might be more relevant. But also the risks in populations at low risk or low prevalence of disease where the positive predictive value might be quite low, and so the risk of a false positive. So both false positives and negatives are already described on there, but the panel felt like maybe they should be described in more specific detail for different populations. And also one of the risks that didn't appear to be included was the risk of an indeterminate result, because that did have clinical ramifications and people have had to deal with that in different ways. And then finally, the risk of inappropriate use, which you specifically mention in the risks that are listed, the risk of incorrect use. But again, it's probably worth listing the risk of people ordering this test when this test is not appropriate for that particular patient. So, and one of the risks that specifically could be called out was when you have an incorrect result that could lead to treatment delays for other diseases. And so perhaps that was worth calling that risk out as well. Okay, and so that was pretty much all of the list of comments that we had about the risk assessment as provided. Dr. Stencil, was that sufficient?
Yes, no follow-up. Thank you to you and the panel.
Great. So let's go on now and discuss mitigation, which I think people, we're all in that boat where we kind of talk about both at the same time because they go together for us. But let's discuss the potential mitigation measures you'd like to see in place for assays that are classified in class 2 and special controls the FDA should consider to mitigate those risks that we've just been talking about. Dr. Prokop?
Yeah, thanks, Gary Prokop, American Board of Pathology. So these tests are particularly susceptible to pre-analytic variations. And the companies currently do a great job on laying out exactly how the specimen should be drawn, how the specimen should be processed, etc. If those aren't done right, those lead to your indeterminate results, etc. So in addition to appropriate patient population, which I agree with completely, is all of the pre-analytics that are currently in place. We would want to see those pre-analytics held to that same degree of stringency with the downgrade. So I'm supportive of the downgrade, but we've got to keep that high level of pre-analytic stringency. That's all.
Thank you. Anyone else have any mitigation or special controls that they'd like to see help with the topics we've just discussed? I wonder too, I wonder too if in the assays, maybe we should be encouraging manufacturers to describe results as invalid rather than equivocal, because they really are not interpretable for a clinician. And so even though the test generated a result, the result's meaningless. And so wouldn't invalid mean more so that somebody wouldn't do what you just described, which is a little bit of a nightmare. But Dr. Cotton, go ahead.
I think invalid would be great terminology because that's really descriptive. That's not borderline. It's just invalid. That's more crisp.
Or uninterpretable, something.
Yeah, something. Those are much clearer. As far as mitigation, I'll just describe what I do, which is sometimes. So I see everyone who's undergoing organ transplant at Mass General in a pre-transplant evaluation. I see almost everybody. So it's hundreds of patients a year who are going to undergo immunosuppression. And we have a pretty significant international population, so we have a lot of latent TB. So I see a lot of people that when I eyeball them, they have a lot of TB risk factors. And sometimes one test is negative, but they have a lot of old granulomas or just like a huge history or whatever. And then I actually use the opposite test. So I switch between T-spot and quantiferon. I don't think that the FDA would want to get into the business of recommending that, but that is a mitigation strategy that I personally use. I used to use skin testing, but again, I've moved away from it just because it's like, and after the pandemic with virtual visits, it's impossible to do. I don't know if other people have comments on that as a mitigation approach. I always say that I take the positive test. Like if I have two tests that are contradictory, then I round up, especially in the setting of risk factors. If somebody has 0 risk factors, usually they haven't taken a detailed enough history to elicit the risk factors, but that's just what I use as a mitigation strategy.
But that's not uncommon. I mean, and we see that even with HIV, not with negative results, but with positive results, where we say that you need to confirm this with a different assay. So I don't see that that's a limitation that's unusual. But in this case, it would be negatives. Maybe if sufficient clinical factors dictate and you get a negative result, you should consider retesting with a different assay. I mean, I don't see why that couldn't be a labeling thing. Dr. LaHose.
I think we have a similar situation as Dr. Cotton here in Texas, with a large proportion of transplant candidates that are born in Africa or Central America and South America. And I guess there's different ways in which this could be approached, but again, it's not a limitation of the test per se, but I think about the pre-test probability. For example, I was born in Peru, and that is the country with the highest incidence of TB in Central and South America, at least when I used to practice there. And if somebody is born there, probably 90% of the population has laboratory evidence of LTBI. So even with a negative test, I may decide to treat them for latent tuberculosis prior to transplant. Another scenario is that there's calculators where it actually factors risk factors, age, specific states within different countries that have high endemicity, X-ray findings, and so forth, to calculate what is the post-test probability of a positive test like this or a negative one. And I factor all that into deciding whether I will treat for latent TB despite the test being negative, in particular in a transplant patient where the risk of developing active TB could be up to 50 times higher than that of the regular population.
Thank you. Dr. Petty?
Kathy Petty, HealthSpring Global. I agree with Dr. Cotton and Dr. LaHose. I also want to caution all of us when we're not dealing with immunocompromised hosts. Many times these IGRA tests are being used as screening tests in extremely low risk populations. They come across as positive. You do an exhaustive history. You can't even elicit, yeah, I was standing in line in Disney World for 6 hours and someone was coughing behind me. And then you repeat the test within the same class, meaning another quantiferon, for example, and then it's negative. And so perhaps in the labeling, we wouldn't necessarily be so prescriptive that it would have to be a different quantiferon versus T-spot, but certainly important to consider. But on the other hand, I've just had patients that have flipped back. It can be a mess in the general population, I guess is what I'm saying. So in the appropriate clinical context, once again, needs to be emphasized. And confirmation by the same or another method could be perhaps a mitigation strategy in the labeling.
So I'm hearing, I just want to make sure I'm hearing correctly, but we're in some populations, based on a risk assessment, negative results would need to be confirmed, while in other populations, because again of a low risk assessment, positive results would be. Am I getting that correctly?
Yeah, I guess the way that I think about this is that if the pre-test probability of latent TB was exceedingly high and you have a negative test, I'm not sure that the second negative test is going to take you to a post-test probability of 1% if you start with a pre-test probability of 90. In the opposite case, if you start with a pre-test probability of 0.0001, even the positive test is not going to take you with even a pretty reasonable positive likelihood ratio. It's not going to take you to a post-test where it's going to even approach 1%. So I'm not sure that in either extreme repeating the test is necessary. It just needs to be contextualized that depending on the risk of the population, you need a positive test doesn't equal yes disease and a negative test doesn't equal no disease. It's just a math issue of pre-test diagnostic performance and then post-test probability.
So should we say something more like, without including positives or negatives, but consider retest based on pre-test positive probabilities? Yes, no, I lost Dr. LaRose.
Yes, just pretest probabilities, because it could be negative or positive.
Right, that's what I'm saying, yeah.
Yeah.
Pretest.
I guess implicit in that is I'm not sure that also a second test is necessary if really you start with a low pretest, even if the test is positive. You just say, okay, well, we're going to stop there. Additional tests are not going to take us to a place where we're going to make this a high post-stress probability of latent TB, and the other way around with the negatives. So what I'm saying is I'm not sure that the additional testing is necessary in every single scenario. There will be some in which it may, but not in all of them.
Right, which is why the word consider I think would be important, but regardless, you'll correct me if I don't capture it when we get to the end. Dr. Moore.
Yeah, in thinking about this, I'm in a state with a large rural population, and most of the TB tests that are done, that is the IGRAs that are done by non-physicians. They're done, in terms of employee health or other work-related issues, educational systems. And so I get this, when they're negative, I don't get the calls, but when they're indeterminate, I get the calls, or and very often they've already been repeated. I guess my recommendation regarding Dr. LaHose's statement is I concur with what he says, and that is that my view would be to try to discourage as much as possible retesting, and really sort of leave that out of the discussion, other than to say, consider if for patients who have a high pretest probability, who have a negative test or an indeterminate test, then additional investigation may be worthwhile, or something to that point.
I guess the reason I keep bringing this back up is this something we feel like there could be a mitigation put into labeling or in the limitations section of a package insert, such that it was more clear to clinicians how to act based on these test results? Or do we feel like there's not enough information? Because I think that's part of the piece that will drive whether this can be changed from a class 3 to a class 2, is if we feel like there's enough information to create the right limitations and or labeling or special controls. So think about that, and we'll come back to each of you as needed. Dr. Cotton?
I think Dr. Moore's points are really well taken. I don't want to create a situation where we're routinely recommending it. I just meant in maybe 1% of the patients I see do I use additional testing. So I didn't mean to open a can of worms. I'm going to put into the chat, there's this great online tool, this TB calculator. I don't know how many people have used that, and I don't know that the FDA would be in the position of, recommending use of this, but it is brilliant. And for risk mitigation, this has been super helpful in my clinical use. It's also something that helps a lot with patients to buy in, because many patients don't believe quantiferon results. Like they say, oh yeah, I had a BCG when I was 1 year old and 65 years ago or whatever. So it helps a lot with overall understanding, and I think helps with risk mitigation. I don't know.
And that's showing up in the chat for anybody who would like to click on that. Okay, Dr. Gerald?
Yeah, okay. There were a few things, and I'm trying to make sure that I get back to all of them. So I will say that the tests that are approved now do have, statements to, the effect about the potential for false positive and false negative results, and how, even in the intended use statements, they really are only supposed to be used in conjunction with, these other, history and other test results. The other thing that I will mention is that, so I think some of the conversation gets probably a little bit beyond what we would put into the individual label of a specific test. For a manufacturer that has decided to have an indeterminate or borderline result, then they clearly need to have very explicit instructions on what someone should be doing, whether it's retesting, how you retest. But then once you start getting, larger than that in terms of like your clinical algorithm of what other things you should specifically be going to next, and because as has been discussed, it's going to differ with your population, with your pretest probability, exactly what would be your next step. That's kind of more outside of our realm and more in, the kind of like larger professional society guidelines and, like ATS, IDSA, this kind of broader picture of how you deal with these tests in general, versus I think where our labeling would be more specifically, to address things that we've observed for the validation of that individual test. I mean, there are some general things, but they're I think more general than some of the discussion that we've been having. And then lastly, which is, I think really beyond the scope of this discussion today, when we talk about these indeterminate, borderline, equivocal results, it is something that we have a conversation with sponsors about, but the idea is that there is a category where it is a valid result that still provides some information, right? So when we know, for example, in these IGRA, so, some percentage of these indeterminate results are because, the sample was mishandled. So this person could be positive and the sample was mishandled and it's like indeterminate. So I think, we can always have the discussion with the sponsor, like, it should just be a straight qualitative and these should be invalid. But there's still, it doesn't take away the impetus for them to include, this sort of category in some circumstances. But we definitely do suggest that they have more simple interpretations when they can. I think that was everything that I wanted to comment on.
Dr. Winston.
Yeah, I just wanted to mention, because some of the discussions here remind me of what we have currently in the cervical cancer prevention arena, where we're very engaged in guidelines development, and recently there were new technologies approved by the FDA where the statements really were according to clinical recommendations or clinical guidelines, and then these guidelines developed by 20 or more clinical societies across the US then give all these detailed recommendations on how to use these tests, because there are a lot more applications in the screening and management realm than anything that could be covered in the indication. So I think there are many examples where this kind of where there's a very focused indication, but then a reference to clinical guidance that can then really expand on that and can have a lot of these discussions that we just had. And I think that works pretty well in some areas.
Dr. Beavis?
Yeah, so I don't know that this is entirely on topic, but I've just got to say, as a pathologist, to hear that we're testing somebody who is a low pretest probability and we're going to ignore a positive result if we get it. Sorry, but I think all the pathologists in the group are just sort of nodding, why are we doing the testing? And screening in the United States is generally, it's a low prevalence group. A lot of the screening that used to happen, I used to be screened every year, but, those recommendations have backed off. So we have a test that's not 100% precise, just given the requirements of handling, given the nature that, it's requiring cells to produce things in response to something. And so we're using a test that's very different from some of the other tests that we've talked about today in the context of, a low prevalence in the US. And so, I just want to caution the group, we can't be looking for perfection here. The circumstances are just very different, both with the kind of test and the prevalence in the United States.
Other points that people would like to make about potential mitigation strategies or special controls? Dr. Ng?
Yeah, thank you. I want to jump on Dr. Beavis's bandwagon. I do agree everything she stated. I also separately want to make a comment to a laboratorian. Invalid and indeterminate are different things.
They are.
And yeah. So I understand the clinicians here not knowing how to use indeterminate, but I would really caution against substituting invalid for that. They're quite different. So thank you.
Yeah, and I think as I thought about it, I thought uninterpretable might be better because ultimately, they really are uninterpretable. But regardless, I think that maybe it's time that we all sit down, both laboratorians and clinicians, and think about what language we've used historically and what language we should use going forward that clarifies things. But that's probably beyond the scope of this meeting. So have we covered all the topics that people think are potential mitigation strategies? Seeing Dr. Beavis, one more?
Yeah, sorry. And just to clarify from what Dr. Ng was saying, perhaps in the package insert could be explanations for what indeterminate means, the couple reasons that could cause that kind of result. Whether it's specimen handling, whether it's something in the nature of the specimen itself, the patient specimen, what are the different reasons? And I think that could help in the interpretation of that result.
Yeah, I think that's possibly true. Okay, I'm going to try to summarize. Oh, Dr. Cotton.
Thanks to Dr. Beavis for that comment. The problem is that nobody reads, on a clinical level, am I allowed to say on this call, nobody reads the package insert. So I love a package insert personally. I have a whole bunch, I have dozens of PDFs, but so that's a real issue. Part of what to me is one of the biggest issues here, and it's true with 1,3-beta-D glucan and galactomannan testing and all kinds of things. Clinicians just want the answer. They don't want to have to talk to the patient, take a history, interact, think about, they just want the test. Is it positive or negative? And for me, that's been a real philosophy change as we've moved towards computer-based medicine. So I'm not mitigating anything. I'm just saying, for me, it's actually really problematic and is part of the whole situation. I think when we talk about including it in the package insert, if there's a way for computers to actually tell the clinician, more of an answer, that would be risk mitigation. What does indeterminate mean, or what does a negative test mean?
And of course, there is a way. And so we can, as laboratorians, we can put that in the results so that people see that. And I think that, it just depends on, we try to keep the results streamlined because people then tell us that there's too much language on there and I just wanted yes, no. So we can't win. It doesn't matter. So,
no.
Dr. Ng.
Thank you.
Yeah, thank you. I just want to emphasize, yes, we hard code the interpretation linked to the result. And I think we only have 100 character limit. So we get our best editor to take that paragraph, distill it down to 10 words, with no more than 2 syllables.
It's like writing an informed consent, isn't it? So can we get to the 6th grade level? Well, all right. So with all that, I'm going to try to summarize. This one's been a really interesting conversation. And I think that because it is a slightly different type of test, and I think in part because it's an indirect test, I think that that puts our thinking caps on a little bit differently. We talked about one of the things that I wrote down that I think, even though we didn't come back to the specific language here, I still think it comes back to there probably needs to be some sort of labeling or limitation that includes what can go wrong if the pre-analytical steps are not accurately followed, because I do think that that is one of the reasons you do get those indeterminants or whatever word it is with that particular assay. We talked about having labeling and limitations that were sort of specific to populations, whether that be populations with active TB, populations that are immunocompromised, or populations that had low pretest probability or high pretest probability. And so, you know, that's something to think about. But I have to say that amongst the panel, we did not actually ever achieve consensus on what we thought that labeling should be. And so we're not being very helpful to the agency in terms of making a recommendation about that, but just something I think that we've clearly identified a point that we think is a point of weakness that needs to be addressed at some point. And I think that there were no real calls for stringency any different from the class 3 level of stringency for sensitivity and specificity, but that we have enough data to understand how the assays that currently are marketed as class 3 assays are working, and we think that those performance characteristics are useful clinically. And so if we have special controls that use that same sort of range of sensitivity and specificity, that will probably meet the needs of providers. Everybody okay with that? Anything I left out? Okay, so now we can go on. Oh, Dr. Stenzel, did you have any additional things that you need to clarify?
Thanks again to the panel and you. No follow-up questions, but one follow-up comment to Dr. Cotton. I really appreciate that you read the IFU package insert. We spent a lot of time on that, and there is so much incredible information in it. So thank you for that positive comment.
I would like to say that I often have the package insert in lectures I give. Like I copy and paste it with some art. So I am an aficionado. I think this is a safe space to acknowledge how much I love the package inserts.
You're an honorary
It is
FDA
nice to hear
member.
that. It is nice to hear that, yes. So it's very funny. Okay, I think we can go on to question 3. And question 3 is essentially the panel deciding if we have opinions about whether this product and other assays, not the specific product, but this type of assay, could be reclassified from class 3 to class 2 based on the information we have currently about what the risks and mitigations are that could be applied to these types of assays. So with that, I'll open it up for discussion. Does anyone feel that we don't have enough information to reclassify these? Let's start with the negative stuff. I saw a hand up and it went away. Okay. Dr. LaHose, did you have a comment?
Oh, no, but now that you're saying the
It's fine.
Yeah,
we give everybody a chance to be negative. We can give you the first stab at positive.
Oh, I think based on the current data, I will be supportive of unclassifying.
Dr. Pereira.
Yeah, Marcus Pereira here. I agree. I would support moving to class 2.
Dr. Moore.
Yeah, I also agree. Level 2 with special qualifications would be the way to do it. I mean, we really don't want to lose the assays, which are critically important, even though their use and interpretation is often nebulous.
Thank you for that. Dr. Cotton.
I also agree with downgrading to a 2.
Dr. Winston.
I agree with moving to class 2, and I think there's a great opportunity to develop like parallel recommendations from the clinical community.
Dr. Moore, did I skip you, or is your hand just still up?
Sorry, left my hand up. Thanks.
Okay. Mr. Spring.
Yes, I also agree with the reduced classification of class 2, and at a prior company, I will add that we made a decision not to pursue such a test because of the barriers that Tim talked about earlier. So again, agree with the down classification.
Dr. Caliendo?
Angie Caliendo Brown. Yes, I also agree with down classification.
Thank you. Dr. Stencil, your hand's up.
Yeah, a couple of comments were made about being worried about losing somebody. Any of the tests on the market. I didn't want to scare anybody. I don't have no knowledge that there is any risk of losing one of the two tests. I just brought it up that there is value in down classification other than getting more tests on the market potentially. Thank you.
Thank you. Dr. Ng?
Agree, thumbs up.
Dr. Blumberg?
Agree. This is a little tougher just because there are fewer standards to compare to, but more urgent to do it, I think, because of that.
Dr. Beavis?
I agree.
Dr. Walker?
Dr. Walker, I agree.
Ms. Schwartzoff?
I agree, and I'm hoping that with this reclassification, it'll bring new people to the table that might be able to solve some of the issues that we have with the current tests.
Perfect. Dr. Petty, it looks like you get the last word.
Kathy Petty, HealthSpring Global. Yes, I agree.
Okay. I will sum this up for you. The panel is in overwhelming agreement that these types of tests should be considered for down classification from class 3 to class 2, and that we have sufficient data for you to take action on moving that forward. Dr. Stencil, did you need any more information from the panel on this topic?
No, thanks for asking, and thanks to you and the panel.
Oh, wait, I have a script and I have forgotten entirely. Okay. Okay, at this time, the panel is going to hear summations, comments, or clarifications from the FDA. Oh, wait, sorry, I have to go back to customer comments. I apologize for this. I got too comfortable with what we're doing. Okay. At this time, I would like to ask our representatives, Dr. Roblina Walker, our consumer representative, Mr. Bradford Spring, our industry representative, and Ms. Jennifer Schwartzot, our patient representative, if they have additional comments about any of the 3 topics that we have covered today. I'd like to start with Dr. Walker.
Hi, Dr. Roblina Walker, consumer representative. I don't have anything extra to add. I am in agreement with every thought and sentiment that has been shared from my panel members. Thank you.
Thank you. Mr. Springer?
Yes, Brad Spring.
Sorry.
That's okay. It's been happening for my life. No, I completely agree with all the panelists' comments and the decisions that were made today, or the recommendations, I should say. I have nothing else to add.
Thank you. Ms. Schwartzot?
I also agree with what everybody has been saying. The more options we have, the better. The more we can get care to the patients. Thank you.
Thank you. So now we will go to summations, comments, or clarifications from the FDA. Dr. Stencil, you have 10 minutes.
No worries, I don't think I'm going to take 10 minutes. This has been a highly productive, very engaging, very robust discussion today. And from our point of view, we're very grateful for all of this. To summarize, except for the hepatitis B surface antigen quantification, we have, I think, solid support, and sometimes unanimous, for down classification from class 2 to class 3. So thank you for your contributions today.
And with that, I would like to thank the panel, the FDA, and all of the open public hearing speakers for their contributions to today's panel. We will meet again tomorrow, starting half an hour later, just to make sure your clocks are appropriate. So with that, this meeting of the Microbiology Devices panel is now adjourned. Thank you, everyone.